This conference will now be recorded.
Well, since I hope everyone is here and we have a lot to cover, we really should begin today's webinar. And I really appreciate you all for attending the webinar today. And a very good morning, afternoon, or evening to one and all present here virtually with me today. Let me introduce you all myself. And my name is Jessica Reeves, Conference Secretariat at Coalesce Research Group LLC, and I'll be available for any of your queries or questions throughout the webinar to help you out technically and organize the webinar successfully. For the next 5 minutes, please allow me to give a brief description about our organization, Coalesce Research Group, and also the further instructions about the GoToMeetings app or the website. So, Coalesce Research Group is very delighted to welcome you all the speakers, supporting associations, and students and the delegates present virtually with us today, with the aim mark the scientific excellence by facilitation, conversations, sharing knowledge. Coalesce Research Group follows through the objective of setting standards and delivering excellence in the research among the researchers, academicians, and industries. Promulgating the research through conducting events and online webinars from various fields of science, especially from stream of medicine, science, engineering, and technology. Within a short duration, we have succeeded in creating a platform for developing a strong professional network with the people across the world from their respective fields that they haven't connected in a while. As a global scientific event organizer, we plan and carry out international conferences, meetings, seminars, symposiums, and workshops, providing the best delegate experience, excellent networking opportunities, conversations leading to innovations, learning new opportunities, lasting working relationships, and professionalization with the professionals around the global network. Just recently, we have also started our own platform for open access peer reviewed scholarly literature where all the content is freely available without any charge to the reader. We aim to share the new recent advances through scientific publications, which will help in minimizing the borders and contributing the use of research works of eminent authors, scientists, researchers, research scholars, students, etc., across a broad spectrum of medical, clinical, pharma, and engineering scientific disciplines and its subspecialties to promote the idea which will guide the development and facilitate the use of scientific information and ensure its quality, and also will be connecting link between the researchers and scientific community. We are committed in ensuring that our journals are disseminated worldwide so that the researchers are able to access our content wherever and whenever they need it. As being the very new journals, we have coined up some interesting journals and have also formed the editorial board, and we will be very glad if any of our today's attendees, the speakers, students, or delegates could extend your support by checking the journals and will be interested in publishing an inaugural issue at any of the journals according to your area of expertise. We will also send you an email with all the details about the journal after the commencement of the webinar, and do take your time to check the website. And finally, I would like to thank the attendees and participants of today's webinar entitled Webinar on Applied Microbiology and Beneficial Microbes, and hope you enjoy the webinar and gain knowledge from the sessions today. So we also hope your participation will contribute to new research findings and that will lead to new collaborations and research initiatives. Before we start with the sessions, I guess some of you will be new to GoToMeetings app. So let me just explain you some basic tools required for your presentations today. So on the GoToMeetings app or web page in front of you, you can find four icons at the bottom center, that is mic, camera, action, sorry, mic, camera, screen, and end. So coming to all these things, so it's mic, that is click on that to mute or unmute yourself. And I request you all to stay muted while our speakers are presenting their research work so that it doesn't cause any disturbance to them while their presentation. And coming to camera, you can use this button to share or unshare your webcam. And if you have the option to share the webcam, you can do it, and it's completely your choice and there is no compulsion. So the next one is share. So you can use this button to share your screen during your presentation. And this is the main part. So during your presentation, after I call upon your name, from my side, I will have to make you the presenter, and only when I give you the permission, you will be able to share your screen. So once I make you the presenter, you can click on the screen button, that is the share button, and you will find a pop-up window to share your screen. And the last one will be end, and this is to leave the session. So again, on the top center, you will have the options to change your display settings. At the top right corner, you will have three icons, that is people, chat, and settings. In the chat option, you can actually send me any message or message to me or any other participants. And for the settings, you can use this button to check for the settings of your speaker and microphone. So as mentioned, kindly remain muted during the presentation so that the presenters don't get distracted or disturbed. And for any questions you have, you can ask the questions, or you can send your questions in the message box after the session completes, that after their talk completes. So for any questions regarding the GoToMeetings app during your presentations, I'll be there to help you out, so nothing to worry about it. So let's just not take much of your time. And finally, I'd like to request you to please give us a brief introduction about yourself before starting the session so that we can know you better virtually. And now let's move into our sessions. And our first session will be from Dr. Margaret Olive from NPO Veggies Cultivation, Estonia, and on the topic Effective Microorganisms Improve the Growth and Quality of Vegetables. So let me just make you the presenter. Yes, Dr. Margaret. Kindly please click on the share button to share your screen. Yes, your screen is visible to us.
Okay, no. Okay, I can't. One moment, I try to. Like this. Okay. Hello, my name is Margit Olle. Firstly, to introduce myself, I have a PhD from Agricultural University of Norway in 1999, which is now Norwegian University of Life Sciences. Then I have had postdoc period, and I worked 12 years in Estonian Crop Research Institute until last year, and from the beginning of this year, I founded the NPO Veggies Cultivation, which I will show one slide of in the end of my presentation also, so you will get a little clue about my new company. But let's start with today's presentation, and the title of my presentation is Effective Microorganisms Improve the Growth and Quality of Vegetables. And presentation outline. Short introduction, what is EM? open land vegetable trials with EM, greenhouse vegetable trials with EM, bokashi technology experiments, soybean open land trials with EM, soybean greenhouse trials with EM in agriculture on very shortly, and overall discussion points. Effective microorganisms. You can see here EM logo on the left-hand side and how it is solved on the right-hand side. EM technology was developed at the University of Ryukyu Okinawa in Japan in the early 1980s by Professor Dr. Teruo Higa. EM has been introduced and is used in more than 140 countries in the world. What is EM? EM is effective microorganism. It is produced through a natural process of fermentation. It is a combination of beneficial, naturally occurring microorganisms. It is a liquid concentrate produced from cultivations of over 80 varieties of microorganisms and is a patented product. Most outstanding feature is it includes anaerobic and aerobic species of microorganisms coexisting in the most beneficially productive manner, not chemically synthesized or genetically engineered, contains microorganisms from different groups. EM consists safe and human-friendly microorganisms, which are lactic acid bacteria, photosynthetic bacteria, yeast, actinomycetes, fermenting fungi, and many more. Coexistence. Lactic acid bacteria and yeast are producing organic acid, which are used by photosynthetic bacteria. Photosynthetic bacteria are producing nucleic acids and vitamins, which are used. The concept behind it. In the soil, in normal conditions, we find 5% of beneficial microbes, 5% of harmful microbes, of opportunistic microbes. What is happening when we add EM in soil? Opportunistic microbes will be working as beneficial microbes and their percentage will increase a lot. Beneficial influences of EM. EM promotes germination, flowering, fruiting and ripening in plants. EM improves the physical, chemical and biological environments of the soil and suppress soil-borne pathogens and pests. EM enhance the photosynthetic capacity of crops. EM ensure better germination and plant establishment. EM increase the efficiency of organic matter as a fertilizer. Different brands of EM are applied as follows: inoculation of EM into the soil, spraying of EM on leaves, soaking seeds in EM, EM irrigation, fertigation, insect control. Open land vegetable trials with EM done by me. We had a scientific project from Estonian company called PRIA, and during this project, which lasted from 2012 to 2014, I made some open land and greenhouse trials with vegetables and soybeans. Firstly, about open land trials with vegetables. We tried cabbages and beetroot and sweet. And here you can see that the yield of beetroot and the yield of sweets were increased using EM application, while the yield of Chinese cabbage and yield of white cabbage, those of cabbages, didn't show statistically different results. Storage loss. Storage loss was less in EM application with beetroots, and storage loss was also less in Chinese cabbage and white cabbage. So cabbages showed very good improvement in storage by EM application. The content of calcium in open land crops. It can be seen that after using EM, the increased. Also, after storage, content of calcium in white cabbage dry matter increased. Also, it can be seen that the content of calcium in Chinese cabbage dry matter after harvest increased. Chemical content in beetroot, Chinese cabbage and white cabbage. You can see here that sugar content increased using EM treatment. You can also see here that the content of nitrates in white cabbage, raw white cabbage, was increased. This is of course not useful because nitrates are of course influencing vegetables negatively in the matter of quality, and also you can see here that the content of C vitamin after storage increased using EM application in Chinese cabbage. There were done several open land experiments also by Estonian farmer Rainer Soosaar from Kõlleste Garlic Ltd. And you can see here that EM treatment increased the yield, while the chemical content was not influenced in winter garlic. Is the influence of EM on summer garlic content of dry matter and calcium content can be seen on picture here, and dry matter was increased in EM treatment while calcium content was not influenced. And we had very good experiments with field peas variety, Estonian variety maize was used, and the yield was increased in EM treatment, and also the contents of nitrogen was increased. This is not good, of course, but the contents of magnesium, phosphorus, and potassium were increased in field pea dry matter. In 2013, I visited New Zealand EM organization in Auckland, and Mike Daly came to visit my trials next year in 2014 with his son from him company. And we went with Agri partner company Argo Cook and to farmer Right Verrev's fields, and we found that using EM helps to decompose soil. As you can see, photo up with EM soil is decomposed when we flow, but the picture down, soil is not decomposed. And with clover root, you can see picture up, use DM on the field, and you can see more side roots and nodules. But picture down, you can see only one main root with very few side roots. Fields of farmer right were also found that oats treated with 1 liter EM per 230 kilogram seeds, variety Ivory had a volume weight by harvesting in 2015 meter, which is quite high. Greenhouse vegetables trials with EM. Here is the most important experiment with tomato transplants because the height of tomato transplants was shorter, they were more compact, so the elements or nutrients went quicker to the top of the plants, and the transplants looked very well. And the widths of tomato transplants were not statistically different, and also the number of leaves were not statistically different, but here you can see again that stem diameter was about 20 percentage larger than 25% larger using EM than without it, and the plant looked very well shorter with greater stem diameter, which you can see here. And the best part of it was that also we got very high nitrogen content, phosphorus content, potassium content, calcium content, and magnesium content in tomato transplant leaves. And here is two treatments with EM on the left and without EM on the right. And also tomato plant from farmer right where of greenhouse. You can see here that one used product was EM, and it got lots of tomatoes from one only plant. Photos of vegetable transplants. Here you can see pumpkin and squash transplants. You can see that the stem diameter of transplants really is large with EM. EM treatment is on the left and control on the right. And also the cucumber plant is shorter in EM treatment compared to control treatment on the right. EM effect on the growth of onion leaves. You can see here that at the end of experiment it proves that the number of leaves in EM treatment is larger, while the length of leaves were not giving statistically different results. Bokashi technology experiments. Introduction. Bokashi thread such as rice bran or wheat bran is inoculated with a mixture of beneficial microbes that flourish in aerobic acidic environments. Bokashi fermentation holds several advantages over traditional compost. Is a closed and anaerobic system. It does not require mechanical intervention, prevents loss of nutrients and greenhouse gases, is acidic and anaerobic and quickly suppresses the viability of pathogens, and fermentation breaks down the complex organic carbon into short chain fatty acids, which are readily available nutrients for of plants. Ferment containing organic matters that are related to the compound product organic range of decomposition stages can see and content. Food waste in the container and set up on the container to collect opacity and the again leaves was great. Is also on the and greenhouse trials. I made some soybean seed germination tests, and it can be seen that soybean germinates quicker by treatment. EM in agriculture, effectively used in soil and foliar applications for regular crop, vegetables, fruits, turf farming, hydroponics. Increases, improves the quality of production. Organic waste can be converted in soil improvement. Waste can be converted into improvement. EM, is it a myth? I think it is agriculture. You can use it on some agriculture. You can use it in animals like cattle, chicken, pigs, fish, mushroom, some methods. The difference between uses of EM in building. EM strengthens the building engineering. Cosmetic soaps, creams, EM called probioticum for human consumption, cancer cure, air purification, bad smell, odor disappears, water purification, lake, seas, ponds purification in Tartu Anne Channel, for example, and so on. And now some words about my new company, NPO Vigis cultivation, and the aim of NP Vigis cultivation is to carry through research and development activities in the vegetable contribution.
Okay, Dr. Margaret, could you please check your network because your voice is completely interrupted.
Of vegetable growing technologies in Estonia, Europe, and all of yes, and good. So that's a word.
Dr. Margaret, could you please check your network because yours it's showing like offline to me. Could you hear me? I'm sorry for
And you
interrupting.
can check it about effective micro.
Let's just wait for 2 minutes for Dr. Margaret to rejoin once again. So in the meantime, I'll just give her a call. Just give me a second. I've just now spoke to Dr. Margaret, so she has a network issue, but she said she's trying. So let's just wait for 2 more minutes and we'll see. And I'm so sorry for the inconvenience. It was the network issue from her side. We're sorry. So let's just wait for 2 minutes.
I'm it. how it's so. So now you can hear me? Go back again, yeah. So yes, but how can I slideshow doesn't want to work now. First, so I can't. From where I could start. Now I don't know what's happening here. Doesn't want to. What we will do? Last slide. Last slide is last slide was that one about yes. I have this lecture in YouTube. I have so you can use actually this YouTube video also. I can send you a link for today's presentation. Yes, I can do that, and we can go on with today's conference because otherwise it's wasting time of others. Yes. That's a very good question. While I'm not so good answering in it, I know a little about the really big chemistry, like metabolites, like biology. I'm more agronomist. While there are a number of microorganisms influencing the growth of the plants, like photosynthetic bacteria. And
So here, Dr. Margaret is really having some serious internet issues, and she said that she will be
bacteria,
sending us the link of her presentation with the YouTube link in the chat. So in the meantime, I have asked her that we have a question, like Dr. Koichi has sent us a question stating which metabolites of bacteria can affect the growth of plants. So, Dr. Margaret, could you please answer that question here to Dr. Kovici? And you're being muted. Please unmute yourself. Again, disconnected. Okay. Okay, let's not waste further time. So we'll just go up with the next presentation in that case because we'll just see what will happen. So yeah, let's move on to our next presentation. And for the next presentation, we have Dr. Abhishek Gupta from University of Wolverhampton, UK, and on the topic production and characterization of biosynthetic hydrogel wound dressing to tackle hard to heal wounds. So, yes, Dr. Abhishek, yes, your screen is visible to us.
Thank you very much indeed, and I hope everybody can hear me.
Yes,
Yes?
we can hear you.
Brilliant. Thank you very much for giving me this opportunity. I'm Dr. Abhishek Gupta from the University of Wolverhampton, and I work as an academic in the Faculty of Education, Health and Wellbeing. The topic of my today's presentation is the production and characterization of biosynthetic hydrogel wound dressings to tackle hard to heal chronic wounds. A quick overview. So basically, I'll cover very quickly a recap on the wound healing stages and the common challenges faced by our wound management team from the clinical perspective. There are so many types of wound dressings, and I will discuss why we decided to go for hydrogel dressings. And again, hydrogels, we could have used so many materials to produce hydrogels, but we decided to go for biosynthetic route. So again, I will discuss why we produce cellulose based biosynthetic hydrogels and how we produce those in our laboratory. Then we wanted our hydrogels to be antimicrobial, and that was the reason we chose silver. And there is a special kind of formulation of silver, which is silver zeolites. Again, I will discuss why we chose silver zeolites. Again, another thing we chose was there was another set of hydrogel dressings we prepared, which were with curcumin. So I'll explain why we chose curcumin and cyclodextrins, and then briefly I'll run you through the characterization for the BC matrix and other two hydrogels that we produced. So without wasting much time, so as we all know, as soon as the injury is inflicted, the priority of the body is to control bleeding. So can you see? So hemostasis is achieved by systemic vasoconstriction and local vasoconstriction that ultimately leads to the formation of platelet plug, and the clot formation concludes this stage. So once the bleeding has been stopped in the exudative stage, the next stage is the inflammatory stage, where the priority of the body is to control infection. So various neutrophils followed by macrophages, they arrive at the wound site, and with their phagocytic action, they try to clear the wound bed or the wound site from the infections. So once this inflammatory stage is successfully achieved, then the wound moves on to the proliferative stage where actual repair starts. So as you can see, angiogenesis, which is the formation of new blood vessels. So angiogenesis takes place, granulation tissue. So this tissue was lost due to injury. Now the granulation tissue is formulated. Epithelialization takes place. So can you see? So the epithelial cells, they start to migrate and wound closure is initiated. So once the dermal architecture is restored, so the tensile strength of the affected area is increased in the regenerative stage. Now, this all looks and sounds fantastic. So, which actually is the case in healthy subjects. So, when the wound healing follows these stages, it leads to healing. But sometimes, due to several underlying conditions or due to infections, wounds can actually get halted in the inflammatory stage, and that can lead to chronic non-healing wounds, as you can see. So in the chronic non-healing wounds, yeah, so the hemostasis or the exudative stage is normal, but the inflammatory stage is prolonged. And you'll be surprised to know that in literature there are cases where the patients have been living with chronic non-healing wounds for years. Imagine the quality of life. And dealing these or treating these chronic non-healing wounds is a big challenge from the clinical perspective. Another challenges which our clinicians or the wound management team they face is the trauma during the dressing change. Because of the obvious reasons, dressings need to be changed. Yes, so depending upon which type of dressing is used, some of the dressing can actually traumatize or affect or harm the healing tissue, which again is a big challenge. Another challenge from clinical perspective is obviously the management team, they decide the regimen. So they decide the treatment regimen for the patient. And in order for the clinicians to kind of assess whether the patient is responding to the chosen or the delivered regimen, they have to monitor the healing process. And because of the obvious reasons, yeah, so they have to remove the dressing, which can again disturb the healing tissue. And finally, with the multiple drug-resistant strains, infections are a big challenge in wound management. Now, I must apologize before I share the next slide, but this is the reality on the ground. So can you see such a nasty type of wounds? If you see figure 3, yeah, that's an example of dry necrotic wound. It's horrible. And if you see here on the other side, so this is a wet exudative wound full of slough. And you must have seen or you must have encountered, so this is a healthy subject and using a gauge dressing, and this wound is not chronic, it's just looks like an abrasion. And when I was talking about the removal of dressing can be traumatic, yeah, so imagine when this subject will be removing this dressing, it can traumatize the healing tissue. Likewise, as I mentioned, in order for the clinicians to monitor if the patient is responding to the treatment, they have to remove the dressing, and as you can see, it can actually disturb the healing tissue. And when the in the regimen, the gauge dressings are used, if they're left for a long time, as you can see, they can get embedded, they can get trapped in the healing wound. And because again, it needs to be removed, and most likely in this case, it's got to be the surgical intervention. And again, that will be traumatic to the wound itself. Now, when we talk about gauge dressings, yeah, so no doubtedly the gauge dressings, which are a type of conventional dressings, they have been in use for ages, yeah, and they have several advantages, to be honest with you, yeah, they are economical. Likewise, they are protective as well. They protect the wound from external environment, yes, and they can be loaded with the healing agents as well. But truly speaking, is that all what we need? If you think yourself, so do we only need this passive approach? How about if we design a dressing that can actually interact with the wound rather than just delivering? Yeah. So based or keeping that in mind, we took an active approach. And when we were doing literature, we came across the concept of moist healing at the wound bed, which was proposed in 1962 by Winter. Yeah. So based on that, we decided to go for hydrogel wound dressings. So our dressings, they have a potential of absorbing the exudate from the wet wounds or exudative wounds. And on the other side, if it is a necrotic, dry necrotic wound, our hydrogels are capable to donate moisture to the dead necrotic tissue. So which means it will work for necrotic tissues or the dead dry necrotic wounds as well as exudative wounds, and maintain optimum microclimate at the wound bed or the wound site. Likewise, I'll explain in the slides later in the presentation, we loaded it with antimicrobials. Yes, so that is how we planned to manage the chronic wounds. So now, as I mentioned, so there are different type of dressings, and the reason why we chose the hydrogel dressings are because they have the capability of creating the moist microclimate at the wound bed, which is proven to facilitate wound healing. Again, when we talk about hydrogels, there are several types of material which could be used for hydrogel preparation, and there are some synthetic material as well, and some chemical reactions whereby we can cross-link the chemical reagents to produce hydrogels, but we wanted to stay natural and green. Yeah, hence the reason we chose the biosynthetic route. So we used bacteria which is Gluconoacetobacter xylanus, and we feed it with the glucose rich HS media. There is a standard recipe for HS media. So we inoculate the HS media with Gluconoacetobacter xylanus, and we incubated it under at 30 degrees for roughly about 2 weeks. Again, depending upon how thick we want our dressing to be, we could leave it for longer or for shorter period of time. So after the set duration of time, we harvested it. And can you see these pellicles? So we got these pellicles, but at this stage, these pellicles are full of the media as well as the microbes, the Gluconoacetobacter xylanus trapped in it. So we purified it. Yeah. So the first boil we gave was in 1% sodium hydroxide, and subsequently we kept on boiling it in deionized water till we get ultra pure, clear, transparent, ready to use hydrogels. Okay, so the reason why we chose bacterial cellulose, again, it is biosynthetic, and as you can see in our experiments, we used conical flask, hence we got these round pellicles. If we had used different shape of vessel, for example, a rectangular or squarish kind of vessel, that would have given us a different shape. So which means we can mold the shape of the pellicles that we produce. Likewise, as I mentioned, we could dictate the thickness as well. Second thing, it is transparent, as you can see here. So it's at the back of my palm and it is transparent. It is ultra pure. When I say bacterial cellulose, it is produced by bacteria and it is cellulose and only cellulose. So if I ask you, when we talk about cellulose, what is the first thing that comes to your mind as a kind of source of cellulose? Would anybody want to share with me? So if we talk about cellulose, what is the main source of cellulose on our planet? It's the plants, isn't it? And plant cellulose contains several byproducts. But this
Like
bacteria,
plant biomass.
that's right, yes, yes. And when we talk about plants, yeah, so plant cellulose contains several byproducts, for example, hemicellulose, lignin. But when we talk about bacterial cellulose, it's only ultra pure cellulose. And to be honest with you, it is so pure that it can be even eaten as well. Okay. And another thing I will show you in the following slides, it has ultra fine network structure. So when we say ultrafine network structure, that allowed us to impregnate it with healing agents, because as only the neat bacterial cellulose, it does not have any inherent healing properties apart from its moist nature. It is not antimicrobial. But when we are talking about chronic wounds, we need some kind of antimicrobial agent. And when we talk about antimicrobial agent, again, we did not want to go down the route of antibiotics, and we wanted to stick to the natural routes. So after literature search, we came across, obviously, the antimicrobial, very, very strong and well-known antimicrobial properties of silver. Silver works against gram-positive as well as gram-negative bacteria. Yes, but the problem with silver is it can be toxic as well. So to tackle with that, and the cost was another issue. Yes, so in order to prolong the release of silver, likewise to bring the cost down so that the dressing could be left on the wound bed for longer, we decided to choose silver zeolites. So zeolites are nothing but the aluminosilicate rocks. So we exchanged silver with the ions in the zeolites, hence we produce silver zeolites. So that way we were able to control, slow down, and prolong the release of silver. Yes, again, I will share with you when we were comparing the results against silver nitrate, we were getting prolonged release with silver zeolites. Now, as you can see, so these are our silver nitrate loaded bacterial cellulose hydrogels. And these ones with silver zeolites, we took two separate approaches. One was we loaded it under static conditions. And as you can see, I don't know how obvious is this one on your end, but it is not very consistent loading. Yeah, so based on that, we loaded it under agitated conditions, and it's pretty much very consistent loading. And that is why we standardized agitated means of loading as a standard protocol. Now, silver, as I mentioned, is very strong antimicrobial. But when we are talking about chronic wounds, we want something to have antioxidant activity as well, because chronic wounds could have very high oxidative stress. And likewise, they are stuck in the inflammatory stage. So looking at all these three things, so antimicrobial, antioxidant, anti-inflammatory, again, choosing the natural products, we came across curcumin. So curcumin is natural. It has well-known antimicrobial, antioxidant, and anti-inflammatory properties. Now, the problem with curcumin is it is hydrophobic. It does not like to dissolve in water. And when it comes to loading, no matter if we were trying to do agitated loading, we were not getting very consistent loading. Hence the reason we use the solubility enhancers. And that was the reason, again, staying natural, we chose cyclodextrins, which are oligosaccharides. Yeah. So the native cyclodextrins are alpha, beta, and gamma. And there are some modified as well. So hydroxypropyl beta cyclodextrin. We tried beta cyclodextrins as well as hydroxypropyl beta cyclodextrins. And these molecules are actually amazing. They have hydrophilic outside, which means they'll very nicely dissolve in water, and they're like donuts, and the inside is hydrophobic, where the curcumin would sit very nicely being a hydrophobic drug. So curcumin sits inside the cavity here. So that is how we managed to enhance the aqueous solubility of curcumin by making complexation with cyclodextrins. And the beauty of cyclodextrins is they are cytocompatible. So they are compatible with our tissues, and they're biocompatible even with our blood as well, hemocompatible. Yes. So the method, again, due to the time constraint, I will not be able to share the complete method with you, but that has been published in Gupta et al. 2019. So we chose solvent evaporation method. So we dissolved curcumin in acetone and cyclodextrins in water. Yes. And we formed the inclusion complex by using 1:1 mole ratio of curcumin and cyclodextrins. So these complexes we loaded into our BC, and we produce the curcumin loaded BC hydrogels. Now, so when we have produced the hydrogel, so we did some characterizations. So we did the characterizations on the neat BC purified hydrogels itself, because we wanted to see how much moisture it retains, or what is the morphological kind of characteristics of our BC, so that would it allow us to incorporate or load any antimicrobials to it. Likewise, as I mentioned, we did the moisture content, and as I was talking about transparency, so monitoring the wound without the need of removing the dressing is a crucial thing. So that was the reason we did some transparency tests as well. Water vapor transmission rate, because once the integrity of the skin is compromised, yeah, there is highly enhanced or more water vapor transmission rate that can lead to dehydration of the skin or the wound. So we wanted to check, once our dressings are applied on the wounds, what is the water vapor transmission rate. Likewise, we tested the thermal stability of our hydrogels, so that in order to sterilize them, can we autoclave them? Yeah, and we got very promising results. We tested their compatibility with blood. We tested their compatibility with different type of cell lines, human cell lines. We tested lung cell lines. We tested brain cell lines. We tested pancreatic cell lines. We tested kidney cell lines. Yeah, and obviously we tested the antimicrobial as well as antioxidant activities. So looking at the morphology, so can you see? So this is our BC bacterial cellulose before purification, and can you see these chaps here? Yeah. So this is gluconoacidobacter still trapped in the network structure. So after it's been purified, can you see it has got a interwoven network structure, and we got rid of all the media as well as the bacteria. Then this is the SEM of silver nitrate loaded BC. As I mentioned, we use silver nitrate as well for comparison purposes. So this is silver zeolite loaded BC, but loaded under static conditions. Can you see? So there were clusters in one end, and there was some empty BC on the other end. So we were not getting very consistent loading. And this is under agitated condition, pretty much very consistent compared to the static loading. And this is our curcumin inclusion complex with cyclodextrins. So curcumin and cyclodextrins are roughly kind of, you can say, irregularly round. But when we make the complexation, they make these brittle plate-like structures. And these actually get impregnated into the BC network, and that is how we manage to load our curcumin inclusion complex into the BC. We did the EDX studies to understand the elemental composition, because of this was the EDX before purification. So it has got carbon being cellulose, oxygen, and along with that it had several other elements from the media. Yeah. So, and nitrogen especially, can you see? So it had nitrogen as well. But once purified, so that confirms all the other elements from the bacteria as well as the media were removed, and we only got carbon and oxygen. So that confirms this is a purified BC. Then we wanted to test our silver loaded BC as well. So again, you can see a big trace or big peak of silver in silver nitrate as well as silver zeolite loaded BC. So as I was talking about the release, yeah, so can you see? So when we compared the release, so we were not getting that much release with silver nitrate, whereas we were getting high release and more controlled release with silver zeolites. And then, if you remember, I was talking about the transparency test. So we did a very, very crude kind of test in our laboratory. So where we used a petri dish and we had text in different color, as you can see. So this is the neat BC loaded with the PBS. Yeah. And this one, as you can see, when it is loaded with silver zeolites, it is not very promising in terms of monitoring the wound without the need for, or without removing the dressing. But when we talk about curcumin loaded BC, you can see it's pretty transparent. So again, this can be dictated because in this one we used 1% silver zeolite, which is way more than what we require above the MIC MBC for various gram-positive and gram-negative bacteria. So if we were to use less concentrated silver zeolites, we could have got better transparency results. Likewise, antimicrobial, as you know, that the aim of our study was to produce antimicrobial dressings. So we chose Staphylococcus aureus and Pseudomonas aeruginosa. Yeah. So, and as you can see, so at 24 hours, we were getting higher activity with silver nitrate loaded BC. So, which shows that it was a burst release here. Yes, and at 48, at 72, and at 96 hours, our silver zeolite loaded BC was giving us higher release. So that confirms that zeolite structure was kind of controlling the release. So probably what was happening was there was an ionic exchange of silver with other ions in the media. Yes, and that is how it was more controlled. So which confirms that silver zeolite loaded BC has a controlled and prolonged release. Can you see at 96 hours? Yeah. So the antimicrobial activity with silver nitrate was very little. Yeah, compared to silver zeolites. So this could be extended even up to 120 hours, which means even the cost of these dressings would be economical compared to other silver nitrate loaded dressings. And likewise, when we tested it against Staphylococcus aureus, yeah, similar pattern. So at 24 hours, we were getting higher activity with silver nitrate loaded BC compared to silver zeolite loaded BC. But at 48 hours, so we were getting pretty much the same. And after 48 hours, we were not getting any antimicrobial effect with silver nitrate loaded BC. Whereas with silver zeolite loaded BC, we were still getting activity at 72 hours. And there was no growth on the discs, 7 millimeter discs, even at 96 hours. So that confirms that our dressings were able to deliver prolonged release of silver, which is an antimicrobial agent. Another thing was we tested curcumin cyclodextrin loaded BC as well. And for this one, we were not getting very, very promising results with the Pseudomonas, but with Staphylococcus aureus, at 24 hours, we got very high or very promising antimicrobial activity. So again, because of the time constraint, I could not share all the results, but I will be sharing the references with you. So in nutshell, or to conclude, we can confirm that the hydrophilic nature of BC hydrogels allows impregnation of healing agents. And in this presentation, I've discussed about loading of silver nitrate, silver zeolites, curcumin cyclodextrin complex, and silver zeolite loaded BC exhibited antimicrobial activity against gram-positive as well as gram-negative bacteria, so Staphylococcus aureus and Pseudomonas aeruginosa. And curcumin cyclodextrin loaded BC demonstrated very strong anti Staphylococcus aureus activity against Staphylococcus aureus. Thank you very much. I hope I stayed in time. Again, because of the full screen, I could not see how long I was taking. But as we are running late, so yeah, I hope I have stayed in time. Any questions, please?
My
Thank
opinion.
you for your presentation, Dr. Abhishek. So, yeah, do we have any questions?
Yes, I want to ask one query from Dr.
Yeah.
Abhishek. Thanks a lot for sir innovative talk on this topic. My one query. Nowadays, sir, black fungus like mucor and blue fungus or like sclerotinia or blue fungus, Aspergillus, penicillium. These are causing different type of mycotoxins. So these wood worms basically caused by mycotoxins and we using like hydrogel. Their mechanism of healing is the similar way. My one query. Another query is sir, what is cotton? Is the cotton fiber, cotton hairs, basically the poorest source of cellulose. So any possibility to exploit these cotton cellulose, fiber-based cellulose for your this hydrogel making products. Thank you.
So if I understand it correctly, so first question is you're asking about the efficacy of these antimicrobials against different strains of fungi. Yeah. So that is your first
Actually,
strain.
different okay,
Sorry,
mycotoxins. Yeah, different fungi produce a different type
microtoxin.
of
Yes.
mycotoxins. Yeah.
Yes. So again, if we can control the growth of fungi, yeah, the earlier we control it, there'll be less or comparatively less release of microtoxins. Yes. So the way, because we are undertaking research on antifungal properties of our dressings as well, and we are not only limiting to silver and curcumin. Again, if you look for our group in the literature, you will see we are working on neem extracts as well. We are working on, our group actually designed a novel approach of following the green chemistry producing silver nanoparticles as well. Yeah, so we are testing the antifungal properties and we have published the antifungal properties as well. Yes, so going back to your question, so if we control the growth of fungus on the wound bed and the early stages, there'll be less secretions or there'll be less production of those toxins. Hence, we will be able to control it. Yes, I hope that I have addressed the first question. And the second question, if I've correctly understood, you were talking about comparison between our bacterial cellulose and the cotton fibers. Yes. Now, in nutshell, if I say our hydrogels or the bacterial cellulose is superior to cotton, the main reason being cotton fibers, they have a tendency to shed on the wound bed. And pretty much, if we talk about cotton fibers in the gorge dressings, that's a good example. So in gorge dressings, even when you are cutting the dressing, you can see that the fibers, they kind of tend to shed. And that is not good for the granulating tissue. Whereas the bacterial cellulose, it's like a mat. So there is no chance of any fibers because it's interwoven. It's an interwoven kind of naturally produced um, network, you can say, yes. And there is no shedding. There is no shedding as you notice in the kind of cotton swabs or even the gauze dressings. So it's way more superior than the gauze dressings or the cotton fibers. Pleasure. Do we have any other questions? Did I stay in time?
Yes, yes, of course. Yes. So, do we have any other questions?
Thank you very much.
Okay. Thanks a lot, and it was really informative and neatly explained, and thanks a lot for that. Thank you for your time.
It's been a
So,
pleasure. Thank you. Thank you to all the listeners and the organizers. Thank you.
thank you. So now let's move on to our next presentation. And for the next presentation, just want to find out if Dr. Hari S. Mishra is available online. I guess he's not available online. So, okay, let's move on to the next presentation in that case. So, yes, for the next presentation, we have Dr. A.N. Tripathi from ICAR, that is Indian Institute of Vegetable Research, India, and on the topic emerging diseases and their management in vegetable crops. So let me just make you the presenter.
Yeah, thank you. Just I am sharing my slides.
Yes,
So yeah, it is shared.
your screen is
Yes, ma'am.
not yet visible. So we're just waiting. Could you
Now,
please? Yes, now it's visible now.
okay.
Mhm.
So good evening, all of you, and I'm very thankful to organizer, uh, Ms. Ilsa, for giving opportunity to present my talk on this particular webinar on applied microbiology and beneficial microbes. It is visible. Yes, I am audible. Yeah.
Yes, you are
So,
audible and your slides are visible.
right. Emerging disease and their management in vegetable crops. Nowadays vegetables, United Nations General Assembly declared International Year of Fruits and Vegetables in 2021. It means vegetables play important role for nutritional security as well as employment generation and income as well as development of entrepreneurship. And in my country during the pandemic COVID-19, and that is the play important role for employment generation for our rural youth and farmers. So, this is very important. Vegetable, India is second largest producer of fruits and vegetables. India produces 191.7 million ton vegetables. That is 16% of global vegetable production in the world. And only we have area under vegetables like 10.35 million hectare only. But what is our contribution in international market? Only 1%, and India is lag behind for processing of vegetable produce, and only we have capacity to produce 2% only vegetables. If you see like livelihood for in India, and more than 11 million farmers income and employment generation comes from these vegetables, and vegetables playing very important role for diversification of our agro ecosystem. If you see like losses, there are two different type of losses, like field losses up to 40% or sometimes more than 40% in the after post-harvest losses during the storage and transportation of vegetables. If we prevent 1% loss, we feed 1 million population. It means it is the great. If we prevent 40% losses of vegetables, we can produce 20% more vegetable production. So, before reporting of different type of emerging diseases in vegetable crops or emerging ecosystems, we have a strong network between laboratory, field, and growers. So we can correct identify the emergence of the disease and what type of management practices and action taken by farmers for better management of the things. So very important for regular interactions, meeting, scouting, and field visits, etc. for correct reporting of emerging diseases in vegetable ecosystem. For these different groups of pathogens like virus, fungus, bacteria, phytoplasma, nematodes, so causing plant diseases, economically important vegetable crops suffered by these pathogens, and viruses among them are toughest after the fungus and bacteria. So similarly, if you see the factors, what is important factor for emergence of the disease? It was found that introduction is one of the most important factors for emergence of the disease. Introduction means what? When the plant breeder develop resistant breeding material, resistant varieties, they require different type of xenoplasm germplasm, so that are moved from one country to another countries for breeding therapy programs. If our germplasm means seeds and cultigens and variety have infection with some particular type of pathogens, so might be chance to long distance transmission of the pathogens from one area to another area, and introduction playing very important role for emergence of viral disease as well as bacterial diseases and fungal diseases. So here why we need emerging disease reporting of emerging diseases. Reporting of emerging diseases and profiling of emerging diseases is very important for management of these diseases and formulation of new tactics for sustainable and viable management practices for better management of these diseases, so we can prevent the losses and we can produce more from less. And other important factors for emerging diseases, reporting of emerging diseases, development of forecasting modules. When we develop forecasting modules in advance, so our farmers prepare accordingly, and we can put our pesticides and other input resources in agriculture, and we can use judicial use of inputs, and we can also cut down the different type of health risk and our adverse impact on environment and soil by using the different type of synthetic and pesticides. So when we report a new disease, so it is very important clearly mention the plant variety, age of plants, name of site means locations, and also owner's name, and that is most important. Action taken by farmers for the controlling of disease. Maybe they are sometimes they succeed, success for management of disease, sometimes they fail. So we can, if you know all things of previous history, that history of problems, so we can formulate the more sustainable, more viable, and more feasible management approaches for the plant diseases. So if you feel, for example, correct diagnosis, the detection and diagnosis of pathogens is very important. So we learn different type of symptomatology, symptoms, what type of symptoms produced by different type of pathogens. For example, fungus, bacteria, virus, and nematodes. These are important category of the pathogens causing build, but phytoplasma nothing causing any build symptom. So we have idea about the symptomatology like build, or maybe food rot, or maybe dumping of, or maybe diagnosis on the presence of the pathogen like maybe sclerosis or maybe mycelia. So on the basis of that, we can correct profile it, and after the correct profiling, it based for effective management of the diseases. So, for example, when the pathogen isolated on different media, so on the basis of their colony characteristics, we identify like particular group pathogen. For example, Trichum is the most important pathogen, say having 8 most important pathogen among the fungal, economically important fungal pathogens in vegetables and agricultural ecosystems, and that isolated on Protein medium, and they look like colony on this. On the basis of their colony morphology and basis of their what is they are producing a special structure known as cerbuli, and the morphology of their conidia, and we have to identify the correct pathogen, so we go for the correct management approach. So aerobiology is also very important for detection of the inoculum in the air when the symptoms not visible in the field. So it is very important for correct development of our forecasting modules for better management of the diseases and assess the race profiling of the pathogens. For example, here also we have different type of pathogens in different vegetable ecosystems, and they are isolated and they identified on the basis of their morphology, on the basis of their conidian morphology, and also they are isolated. For example, fusarium have this type of sickle cell, these are conidia spores, and these are alternaria, alternata, and having this type of uniform conidia, and similarly, Phomopsis vacans causes fruit rot of brinjal having this type of colony and this type of spores. And similarly, the bacterial pathogen now there is more emerging. Second most important economically phytobacterial pathogens, Ralstonia solanaceae, causing bacterial build disease on tomato, potato, chili, brinjal, capsicum, and maybe ginger, or sometimes in banana. So they have been wide host kind of pathogen. That pathogen identified on the basis of their colony morphology, and typical colonies having pinkish center on TZC medium, that is known as Killman's medium. So there are also bacterial pathogens and bacterial bio means races biovars also identified on the basis of utilization of citrate, production of indole, or maybe fermentation of carbohydrates and their products. So on the basis of that, they identify on the basis of that the biovar, and similarly biovar identified on the basis of utilization of sugar and sugar alcohols. For example, mannitol, sorbitol, and inositol. These are 3 important sugar alcohols, and lactose, maltose, and cellobiose are important sucrose. If a bacterial isolates, in case of Ralstonia solanaceae, applied all sugar and sugar alcohol, it is categorized and Bhagavad 3. So on the basis of that, they are having 6 biowars. We identify the biovar. So according to identification of biovar races, we can plan to development strategy for resistance, maybe deployment of disease resistance building programs, or maybe development of resistance, or maybe also explore some chemicals or some bactericides or maybe some other molecules, novel molecules, in molecules and botanicals for vector management according to the race and biovar profiling. And the race identification that is based on disease reaction on the host. If bacterial isolated from organism inoculated in potato, tomato, chili, brinjal, and if they are causing disease symptoms in all seedlings, means also in seedlings, it means that is race one, that is known as Solnicis race. And they are molecular identification also very important for correct diagnosis of the pathogens. So on the basis of that, we can correct identify it, because sometimes of morphological features variable due to different type of nutrients what we can use for their isolation, like cultural media, temperature, etc. So detection, correct diagnosis and detection are required for realistic and reliable molecular diagnostics. On the basis of their uh, differentiation, means homology, we can classify different pathogens, their isolation different groups. On the basis of that, we can link what type of major profiling in particular geographical area, in particular host plants. So we can understand this is the problem of the race distribution. So accordingly, we can make our tactics for plant disease management. And now we have here discussed emerging disease of sowing vegetable crops. Phomopsis fruit blight, that is caused by Phomopsis vacans in brinjal. That is brinjal, very important crop in our country, and that causes 60% fruits due to Phomopsis blight. And sclerotinia also very important pathosystem. And similarly, tomato, and that is fruit blight caused by Phytophthora infestans, is one of the very important emerging pathosystem that is infecting tomato crops in both ripe means green tomato and also ripened tomato. If green tomato infected by this pathogen, so that is not means that is produces aflatoxins or different type of mycotoxins, so that is not fit for consumption and not fit for marketing, and that is causing 15% losses. And similarly, sclerotinia, rhizoctonia, alternaria, also emerging pathosystem causes pre-harvest losses, and that is also means persistence in our cold storage facilities and transportation, and means causes fruit rot. And colotrichum is one of the most important pathosystem in chili and capsicum, that is causes fruit rot and dieback. Bacterial wilt also important pathosystem for this Sunesis crop, tomato, brinjal, chili, capsicum, potato. Other also host plants like banana and ginger is one of the most important host for this pathogen causing some losses. And similarly, here we see what type of different type of pre-harvest pathogens causing different type of diseases. For example, in lead blight. Lead blight causes like 15% losses in tomato, and this early blight caused by Alternaria rot, that's 30%. And similarly, these are rhizoctonia root rot, that's 30% incidence, and that is leukemia and exclusive roadside. And similarly, bacterial pathogen Xanthomonas also causing severe losses in tomato crops. 5% incidence are reported in our Indian scenario and different geographical area and our research farm. And similarly, you see here, Phytophthora blight basically infecting tomato and potato both, and maybe strain A1 and A2. So before we planned our strategy for management, getting more information from inoculum monitoring, what is source of inoculum and how to perpetuate, how to persist. So we can also effectively manage these important diseases. Phytophthora infestans, this is symptoms on leaf and stems on foods. Isolated that pathogen on the potato chips for their correct diagnosis, and if observe, prepare mound, and that mound observed under microscope, so they look like gnats, spongia, and that produces spongiospores, and that is causes mini cycle, that is polycyclic diseases, and very difficult to manage. Nowadays, like metal excels and also use cymoxanil and some other compounds, which also slowing for management of this pathogen developed resistance, so very difficult to get them managed in the field. Farmers so also facing the problem of this. So better understanding of epidemiology is very important for management of this type of diseases. For important epidemiology is very important because it is reduction the inoculum as well as rate of spread of the disease. So yes, and not strategic information can be obtained by these epidemiological models, and we can develop this. For example, Wonderplank rightly pointed out that industries developed chemicals, pesticides, and plant breed-resistant varieties. But plant pathologists validated the strategy for the management, which one of the best approach for management of monocyclic diseases, or soil-borne diseases, polycyclic diseases, airborne diseases, or maybe seed-borne diseases. So accordingly, we have planned to DNA-based monitoring of inoculum, source of inoculum. On the basis of that, what type of pathotypes and what is the prevalency of the strains and diseases, and what is level of fungicide resistance or bactericidal resistance or pesticidal resistance. We assess that. So after we plan and after we recommend particular chemicals in a particular dose, so we can cut down the development of resistance and also cut down the non-judicial usage of different type of inputs. Nowadays, because farmers are aware about what is a health risk of growers, also consumers also know about health risk of these synthetic pesticides. So we can cut down the synthetic pesticides and promote the use of beneficial microbes. And it means if we replace 2% these synthetic pesticides by biocontrols, that is the great impact on the ecosystem, because biocontrol agents are safe, and they are very also producing a different type of growth response in the plants, and also keeping quality and storage quality they enhance. So we can use and we can promote microbials, beneficial microbes in our ecosystem for management of important diseases. For example, these are important ecosystems, so we can test different type of treatments for biological modules that is having beneficial microbes like Bacillus subtilis, Pseudomonas fluorescens, and Deipnomyces, and biochemical modules having a genetic neem-based product, and chemical modules copper, chlorides, tetracycline, azoxystrobin, and we can have a combi module, chemicals and biologicals, chemical and botanicals, integrated disease management modules for their better management of these type of diseases. Earlier, like here we discussed like earlier, phytophthora blight and sclerotinia rot, and it was found that integrated disease management system, when we opted in the field, that minimize the incidence of phytophthora and also sclerotinia rot in only 14.2% phytophthora incidence reported in the field. So it is very important to farmers also integrate all approaches, means resistant varieties, maybe what type of biologicals and botanicals, and if you need, then we can also input into this way of different type of chemicals, means pesticides, bactericides. Similarly, another module, this module, biological module, botanicals, chemicals, etc. Check in coal crops like cabbage, cauliflower, and kale, and broccoli for management of Alternaria blight and Xanthomonas blight. And it was found that biological module, botanical module was the most effective for management of Xanthomonas blight rot in cauliflower and collards. And similarly, these here you see like 11% incidence only alternative blight reported on coal crops in case of biological modules. It means some time of biological modules at par with what is chemicals module. So we can also have opportunity to explore these bioagents and their formulations in our ecosystem for raising of necessary for management of disease in fields, or maybe losses during the storage. And similarly, another ecosystem, most important ecosystem in our vegetables, Pomosis blight, that is caused by Pomosis vacans. Here you see the symptoms of fruit rots. These are symptoms in fruit, and this is mummified food. And this type of symptoms appear during the storage of brinjal. This is also very severe one. So for management of this important pathosystems, we tested different type of varieties that are developed by multinational companies, and it was found that Imperial Long, that is developed by Imperial Agro, and Namdhari S797, and brinjal, because these are most common varieties and most popular, having the largest acreage in the farmer's field, severely infected by these pathogens, and we found that nothing resistant material against the disease. So we can breed resistant material against this disease and also search for new chemicals for the better management of the diseases. For example, here we isolate the pathogen Pomosis vacans and the PDA plates, and that is morphology, and there is some morphology identified as a Pomosis vacans, and there are the Pygnidia and their spores, that is having single-celled and regulated ones. So these are important features for understanding the development of resistance in the pathogen. What is we can use generally use chemicals, so we can search for new molecules and new molecules and some other chemistry for the management. On our institutes, developed different varieties, and it was found that IBBSL 23, IBBL 20, and IBBL are highly resistant against these pathogens. So we can available these materials for our farmers. So it is the cheapest way for management of disease without putting using a family. chemicals and synthetic chemicals or some other chemicals. Some another ecosystem pathosystem like Sclerotinia Sclerotium. This is the very problematic pathogen causing post-harvest losses or even field losses in solanaceous crops and leguminous crops, and we have very difficult to resist against this pathogen. And sometimes also we have different type of molecules, but that synthetic molecules of fungicides not working effectively. Another Polytroticum, that is the Polytroticum capsicum, causes fruit rot of chili, and sometimes when we make like red chili powder, that pathogen mix inside the powder and causes different mycotoxins, and that is identified under this subject colony morphology and PDA, and there are spore on this that is sickle cell spores produced by this pathogen, and that is pathogens and other pathosystems and pathogens isolated Sclerotinia, Sclerotium rhodesi, and also Pneumocystis vacans and different cultural media, and Sclerotinia speciorum and Estonia solani, and emerging diseases of legumes vegetable crops. Sclerotinia speciorum is one of the classical pathosystem, causes diseases in the field as well as storage in French bean, that is Solus vulgaris, or Indian beans, or lobla beans, and some other beans, and Proteicum lilimutinum also important for causing anthropogenic disease in Indian beans. Is to fight up ISI in the partner system, that's why to fight up light in the P, macrofauna, Sclerotium rhodesi, also emerging pathosystems in our leguminous vegetable system. And other than these pathogens, Pseudomonas, Erwinia, also important bacterial pathogens causing post-harvest losses in this crops. Here you see here that is a French bean that known as Razama, that is basically infected by this Sclerotinia sclerotium, and that is Indian beans, Dolichos beans also severely infected by this pathogen. And here you see pathogen isolated under Agar media, and there is Sclerotia look like this type of Sclerotia. If one time Sclerotia farm in the field that is survived to 10 years, we have no mechanism for management of this. So if that Sclerotia is spread with help of seed from one area to another area, means transboundary movement of seed material, so it might be chance to spread there, and very difficult to breed resistance against this pathogen. We have the limited resistance against this pathogen, and similar symptom also observed in the pea, that is the vegetable pea. And another emerging disease pathosystem on cucurbits like gummy stem blight, that is more than 50% of our 50% incidence of all cucurbitous crops like maybe watermelon, or maybe muskmelon, or maybe bottle gourd, or maybe bitter gourd, or maybe pumpkin, or maybe summersquash. Didymilla is the major system of the gummy stem blight, and fruit spot caused by Cercospora, that is now the emerging one, and that is the ESC. We discuss in next slide here. What type of symptoms observed, that is Sclerotinia rot on the bottle gourd, and that is Polytrichum lesnerium intranose, that is reduce the value of marketing value of these food crops, and that is another pathogen known as gummy stem blight. When the pathogen infected the stems, they are produces some secondary gummy-like substances on the base of that identified as gummy stem blight. So pathogen, gummy stem blight pathogen isolated on the media, and look like this type on their spore on the base of spore morphology. They are confirmed with Didymilla browni, now known as Stagnosporopsis browni, and this is the Polytrichum isolated on PDA, and there are several uses on this, and they can identify and use their morphology. Another important pathosystem of this bottle gourd is Cercospora. Cercospora, these are the spores here, and their symptoms on the leaves can managed by using this type of chemical molecules. So we test different type of treatments in the field for the better management of the Cercospora blight. So carbendazim, using of the carbendazim for management of this disease. So 56% incidence was observed on gourd field. But in case of Trichoderma spirillum, that is the beneficial fungicide, that is used for pesticides, and more than 200 biopesticide product basically based on this type of especially that is very beneficial microbes, and using a different way for management of diseases, or maybe plant growth stimulations, or maybe enhancing the keeping quality of different vegetable crops. So that is also have a gap result in the for controlling of this Cercospora part on this crop. So that is very important for integration of our integrated disease system. For example, another emerging pathosystem of bacterial and pathogens here. We discussed that only one pathosystem like Estonia solanaceum causing bacterial build-up economically important solanaceous vegetable crops. That is the if rank, that is the second most important bacterial pathogen, Estonia solanaceum, having 5 bases and 6 by 1, and that is causing the diseases in bacterial diseases in solanaceous vegetable crops, and that is the chance may be spread by by means of seeds, or means of seedlings from one place to another place. So here history earlier, this is the severe problem in hill. like Himachal Pradesh, West Bengal, Odisha, and coastal region of the country. But nowadays, that is spreading from hill to plain and infecting in Uttar Pradesh, where our institute located, Indian Institute of Vegetable Research, and new strains, means Pseudolastomia solanaceae strains, also infecting the field crops, and massive losses in the fields was observed during the last 2 3 years. And that is the disease symptoms are like chili that is variety severely infected by this pathogen, and incidents reported here. And leukemia, stem rot in summer squares, and leukemia, blight, soft rot in cauliflower also reported in different range, up to 20% in summer squares and 50% in another summer squares variety, Cassis subhangini, and similarly up to 75% of bacterial reported in the fields. So these are some photographs where the severe incidence of this pathogen, bacterial pathogen causing bacterial in tomato and brinjal or capsicum or potato. Here you see 100% losses in observe what is that is a very popular variety of bell pepper, California vendor in Hindi district, Himachal Pradesh. So this is the very difficult pathosystems, and management also difficult because having high wide ranges, 5 and 6 survivors, and we have the limited number of bactericides, and those bactericides are available basically not feasible. So it is very difficult to manage by chemicals. So what we can strategy of fire management, we have resistant varieties, the protection or integration of these can be helpful for management of the diseases. In this case, microbes play a very important role. Beneficial microbes like Pseudomonas, Pseudomonas, fluorescence, and their different type of formulations are effective for management of soil inoculum of these bacterial pathogens, and different type of pathogens like these are Xanthomonas isolated from your cabbage, cauliflowers, and these are pigmented colonies, red colors, and different media, and similarly Irvina has soft rot, different media, and Allostonia solution bacterial built in brinjal that is also isolated from Hillman's medium. So different type of morphology on their study. There is a small study conducted for controlling of Irvina soft rot in flower, and that is isolated on different media, and it was found on CRB7, that is Bacillus subtilis, formulation development by Institute, and that is found very effective for management of Irvina soft rot in cauliflower, and incidence of least in case of this only 2% in comparison to control. And Sclerotinia Sclerotium also important ecosystem causes severe losses of cauliflowers. And similarly, different type of molecules, means botanicals, green molecules, and biopesticide formulations, bacterial formulations are available in the market. So these are tested against important bacterial pathogens, and if we found the better efficacy in comparison to chemicals, definitely we incorporate in the ecosystem, so we can replace the synthetic molecules, pesticides, and also promote the organic, and also promote the quality production by using this type of biocontrol agents. For CRB, related trichoderma bacillus subtilis strains associated from IIBR and formulation developed and used against Irvina xanthomonas, and was found that effective against all pathogens. And similarly, different type of botanicals available in the public domains in the markets, and farmers are mostly used for the management of different type of bacterial diseases and fungal diseases. One is spotless, curex, and besides these are also sometimes effective, and so spotless is a better molecules for enhancing the growth, promotion, as well as the management of diseases. And Casugamycin plus copper combi product are maybe copper oxychloride, and also work most effectively against these pathogens, but sometimes develop resistance by the pathogens and the pathogen. So we have need to such new molecules, maybe cleaner molecules, maybe botanicals, or maybe their consortia mode, another, so we can effectively manage the disease. And we have different type of molecules are available in the market, and farmers use. We tested again different type of fungal pathosystem, that is Sclerotinia sclerotium, cause a different type of rots, maybe fruit rots, or stem rots, or maybe in nursery disease, maybe food rots, and so it goes. And it was found that telconazole, hydroxystrobin, telconazole, hydroxystrobin was found effective against management of these type of pathogens. So pyram and telconazole also very effective for management of the pathogens. And one also important pathogen here you see here is spotless, that is obtained from botanicals, and that is very effective against management of all important soil borne, seed borne, these type of pathosystems. And these pathogens what is tolerance tested on fungicidal sensitivity in vitro by using a poison technique. Here we control petiplates, here recommended petiplates. So here least growth means that fungicides tested in vitro conditions are effective against these mycelial growth of the pathogens and different pathosystems here. And we also tested in vitro condition, tested in vitro of different type of bioagents available with institutes are evaluating the markets, or management of microfauna, pythium, sclosium, and Rajatonia, fusarium, and Formosus vacans. Found that trichoderma strains CB2 found most effective for management of these important system diseases by using a dual culture techniques. And sometimes we test the compatibility. For example, trichoderma spirilum available in the market and different formulations. So we can add different type of new generation molecules like deoxystrobin or maybe copper oxychloride. So we can use upon the product. So they are much more. It can enhance the effectiveness and can be, for example, bioprospecting of microbes. So what we have microbes, we enhance their bioprospecting by using the different type of botanicals and chemicals. If they found compatible, they can use in our immune system. So they can cut down the doses of that particular chemicals and also enhance the efficacy. For example, you see trichoderma spirilum is enhanced by using the penicillin or maybe sometime also the image target geosystrobin. So that will be very good. They are eliciting that they are different stimulation in the biological agents like trichoderma spirilum, and they are using dual culture. This is the pathogen and this is the trichoderma. So trichoderma can limit the growth of the pathogen. So on the basis of medicine pattern, more than the trichoderma extends more than 50% of the growth of the pathogens and the mycelial structure or maybe formation of some special structure like sclerosia. So we can use further formulations and the rest for the management of drug. Where we see sometimes we can use a particular chemicals for management of a particular disease. For example, a particular chemical recommended for a group of representative group of the crop. So that label claim means expenses for other crops. But sometimes we have no label claim, chemical molecule, fungicides and bactericides. So how to use in our regular ecosystems and vegetables for the better management. One is if you use not label claim molecules used for management of the pathogens, maybe fungal disease or bacterial disease, and that molecules work effectively. So another question is PSI post-harvest interval. How much residue inside the harvest what is we obtained from the produce? One problem. Another problem is development of resistance in itself in pathogens of fungicides and bactericides. So we can manage by using a different type of fungicidal pyramiding or maybe fungicidal deployment. We can integrate more than two type of chemicals, can be product of chemicals and botanicals and develop consortia. And also we use the rotational system for applying. If you want to use like methyl, so this time we use like elite, positylene, and this time we use trichoderma. So we can change, so we can reduce the development of fungicidal resistance and manage the fungicidal resistance. So in conclusion, this type of history, emergence of new diseases, profiling of emerging of new diseases, documentation of new diseases, better understanding the diagnostics of their pathogens and their variability profiling will be helpful for management of integrated approaches and development of suitable, sustainable, feasible, integrated approaches for management of important disease of vegetable crops. And also we develop different type of resistance varieties in case of the vegetable crops for safe vegetable production and safe movement of vegetables from one area to another area under better diagnosis. If the new pathogens are reported in our product or maybe seeds or some other materials and they move from one area to another area, and if not, we get the phytosanitary and phytosanitary certificate. So very difficult to export that material. So trade also important. So better management of diseases, safe management of diseases is very important in our vegetable ecosystem. If I would like, if some participants want to interact, I would like very happy to interact them.
So thank you for your presentation. So yeah, do we have any questions? Any questions regarding the presentation? I guess there are no questions. So anyways, thanks a lot, Dr. Tripathi, for your presentation. So thank you for your time.
Thank you for giving the opportunity to present in this conference, and I am very grateful to you for learning from different participants, the different angles and different research outputs. Thank you. Thank you.
Thank you. That was so kind of you. So yeah, now let's move on to our next presentation now. So for the next presentation, So it is actually from Professor Hari S. Mishra. So we just missed his presentation. So let's just move back to that. And Dr. Hari S. Mishra from Bhabha Atomic Research Center, India, and on the topic DNA damage response in
Ex-
radio-resistant bacterium.
Excuse
The
me, Jessica.
Yes.
I am Dr. Bushra Munir from Pakistan, and actually, I am waiting from a long time, and I am COVID positive. That's why is it possible for you to take my
Okay.
presentation?
Okay. Yes. Yes. Definitely. Definitely. I can do that. So, Professor Hari Mishra, I kindly request you to.
I am sorry, Professor Harry, because it is difficult to sit for a long time for me.
I completely
Go ahead.
understand.
Go ahead. Go
Thank
ahead,
you.
please. Go ahead, please. I
Yes.
will wait for next talk.
Thank you,
Thank you.
thank you, thank
That
you,
Go
was
ahead.
really
thank you.
kind of you. So,
Sure.
yeah, now let's have the next presentation by Dr. Bushra Munir from Government College University, Lahore, Pakistan, and on the topic cloning, expression, and purification of arsenic reductase gene from Thermus aquaticus. Yes.
Thermus aquaticus.
Yeah.
Actually, I am not, I can only show you for some time that I am Dr. Bushra Munir, and I will switch off my camera. Please don't mind, all of you.
Not a problem.
All right. I am is it shared with you or not?
I'm sorry?
I am sharing my screen, but there is some, I think there is some problem. Have you seen my screen?
Yeah, like we could see it like just now it got closed. Can you please click on the share button once again to share the screen?
All right.
Yes, now your screen is visible,
All right.
but
Thank you very much.
it's
I
just a
am,
plain background.
yes, it is plain because I
Okay,
have prepared in a great tension and all of these. Although
okay,
I am
okay,
vaccinated,
no
but
problem.
still I am having this problem.
Not a
All
problem.
right.
Please do take care and yeah,
All right.
please do continue.
The topic of my presentation is the cloning and expression and the characterization of arsenic reductase gene from Thermus aquaticus. I am Dr. Bushra Munir from Institute of Industrial Biotechnology, GC University, Lahore. The main objective of my research is to isolate the arsenic reductase gene from the thermophiles because in bioremediation we can also use the higher temperature and extreme conditions so that we need the enzyme it can work on that temperature. Arsenic is mainly the arsenic, the main big background of my research is that the arsenic contamination is a global problem, and according to the WHO arsenic is one of the most dangerous substance, and it is the main inorganic. Arsenic is considered as the carcinogen, and it is estimated that about 150 million people are exposed to these metalloids. Excuse me. And the most affected zones for with the arsenic, these are the Bangladesh, West Bengal, and the Taiwan. And WHO recommended according to the WHO, the arsenic level in groundwater, you can say that the recommended limit of the arsenic is 10 microgram per liter, which is about 10 ppm. But in these days, Pakistan is also facing this problem that some areas of Pakistan, like a small village in Lahore, Kalashakaku, as well as Kalalwala, they are they are they are contaminated with the arsenic, and the United Nations Institute, UNICEF, it verified that there is about 10 to 200 microgram per liter of the arsenic is present in their water, and arsenic is released into the water with the different anthropogenic activities, with the natural as well as some geological activities, and the movement of the arsenic is called as the mobilization of the arsenic. And nowadays the biotechnologies, they are making the large number of the broad range of the enzymes, which is known as the microbial enzymes, and which can be used to remove the these are the different oxidases, different reductases, and different isomerases, which can be used for the removal of these metal contents from the wastewater. You know that it also affect on the human body. It produce the body rashes, lungs, it affect on the lungs, urinary bladder, as well as on the kidney. When we talk about the metabolic process of by the microorganism, microorganisms have different mechanisms to remove the metal contents. They have the different extracellular the mechanism. They have different uh, uh, chelating compounds. They have the intracellular transformation, means the microorganism have ability to remove the metal. They have the metals can bind on their surface. It can enter into their into the cell with the help of the different channels, and then there is the different enzymes in the microorganisms which can oxidize or can reduce these metallic compounds. And when we talk about these, there are some proteins which are known as metalloproteins. These are also present in the microorganism. Now, there are large number of the chemical methods, the ion exchange methods, lime softening techniques. There are different osmosis and the reverse osmosis, as well as the filtration processes, which can be used for the removal of these methods. But these methods are very they can use the different chemicals. They are also produce the hazardous metabolites, which can also and they are also very expensive. So we can also utilize the same indigenous microorganism which are present in that environment, because they have ability to cope with these metallic ions. So we can use this microorganism for the removal of these methods. And in this research we isolate the basically the reductase gene from the thermus aquaticus, and we have used this methodology that we have isolated the genomic DNA, then we amplified the desired gene. After that we cloned it into the first into the cloning vector and then into the expression vector, and after that we expressed it and then purified and characterized that protein, and then we utilize it for the removal of the arsenic from the industrial wastewater. For this purpose, the genomic DNA was isolated, and then 559 base pair fragment was amplified from this the DNA, which is used as a template, and we cloned it into the vector that is the PTC57RT vector, and then we transformed this clone vector into DH5 alpha. These white colonies, this shows the gene, a reductase gene, the vector with the reductase gene, and then after that we perform the clonal PCR in order to excuse me, we perform the clonal PCR to confirm our gene, and after that we use the restriction analysis method, and with the help of we cut the cloning vector with the help of the true enzyme, that is the ND1 and not 1. And then you can see this is the plasmid, and this one is the our desired gene. Then we have sequenced this gene, and after sequencing we construct the phylogenetic tree, and we showed that the reductase gene with the other strains of the with the other species of the thermus. Now, after that, we clone it into the PET21A expression vector, and then after that, for this we digest the PET and we clone it, and then we observe these colonies, and then the we like after ligation we first run it we isolate the plasmid, the recombinant plasmid of PET21A with ARS gene, and we run it on the gel, and then we perform the restriction analysis in order to confirm, and our gene was present here.
Dr. Bushra, I'm really sorry for interruption, but I just want to confirm once again, like, are you presenting, like, your slides are not visible, so it's just like you're talking, or are you giving any presentation?
No, my present, but I am sharing my slides.
No, no, only your plain screen is visible to us, not the slides.
Why? What is the reason of this one?
Or
Now,
maybe you might have not given any permissions to the GoToMeetings app. That might be the reason.
wait a minute.
Okay.
Now,
Even now, just the plain screen it's visible.
what is the reason of this one? Everyone, I have given the permission, but wait.
Over there, could you see there is only a go to edit view call window help. Could you please click on that go to? We'll just see if there's anything required.
Wait a minute. I have not seen any message on this one. There is no message on my screen.
Oh, beside the Apple symbol, we could see go to edit view call window help.
Recording, hand, mic, camera, share, and leave. I have only
No,
these.
that is actually not visible to us. Okay, could you please send the presentation to to my email so that we'll see?
All right, I can send.
Yeah, I've sent the message.
All right, all right. I am just sending your. Wait, I am just sending.
Yeah.
Jessica. Jessica, I have just received a message from you, and now it has gone somewhere.
Uh-huh.
Please send me your.
I have sent like the message. Did you see that? Jessica at coalescemeetings.us.
There is nothing on my screen.
No, in the chat
Wait,
corner.
wait, 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 wait. Yes, I have received. Wait.
Yeah.
Hmm. Yes. Yes. I have not received any message from you yet.
No, like, did you find my email ID?
Yes, I am
I have,
sent. I have sent. I have sent.
oh, okay. You've sent, I've actually tried to refresh it, but there's no email coming in. Okay, let me just check once again.
Have you received?
Yeah, I have an email. Let me just check. Yes, received it.
Alright, thank you. Yes, but
Yeah, you can just let me know when to move the slides or everything. I'll be doing that.
all right. May I start from start or
No, no, you could actually tell me to which slide we can go so that we can continue from there because we were listening
all
to
right?
all your presentation.
All right. I am sorry for this,
Or maybe
but
we
I
can
don't know
just
what is
shut
the
it
reason of
here.
this. I think it is due to some internet problem that. All right. Now you can go to the slide number 2020. No, 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 no. Expressions of arsenic reductase. Or may I start from the first? You can go to 323rd slide.
Okay. Yes.
I will send to my these slides to everyone on their email. All right.
Okay.
All right. The
Uh,
expression of arsenic reductase gene. For this we have
is my slide visible? Am I on the correct slide?
yes, you are on the correct slide. All right.
Okay.
The, the expression of the protein was we used the uh, STS page gel to check to estimate the expression of our protein, and we have observed that 22 kDa protein. It is obvious in this gel that you can check that 22 kDa protein is uh, this one is this reductase gene, and also from the we use the both the supernatant as well as the palette to uh, check whether they are present in the uh, whether they are released or they are present inside the cells. And then we have also, please go to the next slide. We have also observed the same 22 kDa protein on the, please go to the next slide. Yes,
Is my
and the
slide
same,
not changed?
yes, the same is
Okay.
present on supernatant, and then we have characterized the arsenic reductase protein, and because we want to see that whether we can use it at different temperature range or different pH range or not. Please move to the other slide, the next slide, the next. And for this characterization, we have used the pH. We use only the two parameters, the pH as well as the temperature. Sorry, the pH, the small p and the capital H, and it is due to it is the typing mistake. And then after that, we have checked that these reductase genes can it can withstand with the broad range of the pH from 4 to the 7. And after this, the because it is mainly the enzyme is the acidic in nature, and after this its level concentration is decreasing with the increase of the pH. Next, please. Next slide. And then the temperature, as we have isolated it from the uh, thermophile, so it can withstand with the higher temperature. From the optimum temperature for its activity is 70 degrees centigrade. Please next. So after that, we use the same enzyme in the same enzyme and enzyme within the organism in which we have transformed it. That one is the DH5 alpha BL and the BL21 codon plus, which is for the expression. And the for this purpose, we have we design our experiment in such a way that we make a setup of three containers, and these containers they contain the 50 liter of the which contain the 10 50 the total capacity of these containers are 50 liter, and they contain the 10 liter of the industrial effluent. Two contain the 10 liter of the industrial effluent, and third contain the 10 liter of the distilled water, which is for the control. Then we maintain the concentration of arsenic in all these containers, which is the 100 mg per ml. And then we have inoculated all these containers with our arsenic reductase contained bacterial strain, and we incubate it for different time intervals, that is the 3 6 3 to 24 hours. And then we uh, after these intervals we took our collect our samples, and we centrifuge it, and we check the metal concentration in the supernatant as well. We check the concentration of arsenic from the supernatant with the help of the atomic absorption spectrophotometer at 193.7 nm. Next, please. And we observed that our our enzyme have ability to reduce about 82% of the arsenic from the medium after 24 hour of incubation. So we can conclude that we can use this reductase gene for the removal of the although it is specific for the arsenic, it is mainly arsenic reductase gene which is present in these strains, but we can also use this the the other reductase genes from the bacteria, even from different microorganisms. We can utilize the bacteria, we can utilize the yeast as well as the protozoans for the removal of the contaminants, because they have the natural strategy to cope with these con- contaminants. So I have also working on the yeast protozoans, and we are also using in the consortia now. And now we want to we want to produce the different bioreactors for the production, because I have a HEC project in which we can establish with the Turkey collaboration, we can establish this bioreactor by using these enzymes to remove these contaminants from our environment. Thank you very much, and sorry for the disturbance, and you can ask the question.
So, yeah, do we have any questions?
Yes, arsenic
Yes, please
is
go ahead.
heavy metals, and
Yes.
arsenic is a heavy metal and very toxic for human beings and animals. And if we use like different type of bioremediation using the what is jalkungi, echinoclora in the water bodies, so that is also problem for removal. So how to best way for management of water bodies where different type of plants that absorb arsenic from water, and how to their disposal of, what is best way of disposal of for botanical materials or biomass have high amounts of arsenic.
Arsenic is, that's why I'm saying that the arsenic is very toxic and it is present in the environment, and it is due to the industrial activities and due to the geological and due to the natural activities it is entered into the environment. But you know that the microorganisms, they are already present in this environment. Uh, if you isolate the arsenic arsenic, you can say that the arsenic tolerant bacteria from that sample which contain the arsenic, it means that this bacteria, this microorganism, it has some ability to cope with this mechanism, these heavy metals. So we have to check that which mechanism there, because microorganism can absorb, and there is some metallic proteins, metallothionein proteins, and there are some other enzymes just like the reductase, oxidase. These are enzymes are present, and we can utilize all these methods, all these mechanisms which are present in these microorganisms to cope with this metal, and we can use it to remove these metals from the same from the same environment. And if this microorganism, you know that the microorganism can be pathogenic, and we cannot produce this microorganism in bulk quantity. So we can, if we produce in the bulk quantity, it means we are disturbing the natural environment. So there is the need to isolate the specific uh, genes or specific operons which are responsible to cope with these metals, and then we can utilize it, we can transform, we can clone it, and we can transform it into the in the environment friendly organism. Even we are working on the plants which can which are able to remove you know that the tobacco plant which is responsible, because we have cloned the genes and we have given it to the botany department. Now they are trying to introduce it into the plant so that they can remove the contaminants, they can reduce the level of these metals from the groundwater.
Thank you.
Hey, can I ask a question to Busra?
Yes,
Yes,
doctor, please
yes, Dr.
continue.
Harry.
Yeah.
I know.
So thank you, Busra. Your name looks familiar to me. I'm trying to recollect that where I have read it, maybe in some paper, etc.
Yes,
Uh,
I have on ResearchGate. I have a connection with you.
I think it looks like that. Yes, I have done something. Anyway, good work. I have just a few curiosity question. Uh, Number one,
Yes,
that this arsenic removal which you did, what temperature did you do?
we are mainly from the industrial wastewater. We use the room temperature, which is about our temperature is in these days is about 40 to 40, 42 to 45. We have used the because in industry it is because they dump all the industrial wastewater into the ponds, and in these days these ponds have the temperature is about 42 to 45, so we use the same temperature. I can't hear you, Dr. Harry.
Professor Hari Mishra, your voice is not audible.
Your
Not
mic is
audible.
off.
You're still not audible. No, not audible.
Hello.
Can you hear me now?
Yes,
Yes.
yes,
Can you hear me?
Yes, we
yes,
can hear you.
Hello?
yes,
Yes,
Dr.
we can
Harry,
hear you.
I can hear you.
Right. So you checked the thermal stability and you found that temperature optima was 70.
Yes,
So do you feel that if you do this bioremediation at higher temperature, it will work more efficiently?
yes, it will. But we can use it into the bioreactors because in bioreactors we can also need a higher temperature. So we can use the same enzyme in bioreactor at higher temperature. Also,
So
it is active from at this temperature. If you check, you can see that this the it can work from 45 to 70 degrees centigrade. Its activity is gradually increasing, so it can work from 45 to 70 degrees centigrade more happily. So we can utilize this broad range from 45 to 70 degrees centigrade.
no, my curious another suggestion is there that if you incubate this enzyme at different temperature and do the activity essay at room temperature or 37 degree, how does it behave?
At 37 degrees centigrade.
No, see, I am trying to bring out when you are
Mm-hmm,
check when you are checking the thermal stability,
mm-hmm,
now that since this enzyme is from extremophile,
mm-hmm.
so they have a better stability at higher temperature. In
Yes.
that in that case, if you incubate your enzyme at different temperature and check
Mm-hmm,
for activity at 37 degrees centigrade or a room
mm-hmm,
temperature, you
mm-hmm.
may get a better picture of thermal stability.
All right.
That
I will do
is
it
the suggestion
because
which
I
I
am
have.
working on this. I can do it.
Right. Okay. Thank you.
Thank you very much.
Any other questions?
Thank you.
Okay, I guess there are no questions. Anyways, thanks a lot for your time, and I'm really grateful for your presentation. And I request you to please to take care, Dr. Bushra, for your health.
Thank you
And
very much.
let's move on to our next presentation then. So for the next presentation, we have Dr. Hari Mishra from Baba Atomic Research Center, India. Let me just make you the presenter. Yes, Dr. Hari Mishra, we have made you the presenter. Kindly click on the screen button. Yeah.
Can you see it? I have done
Yes,
that. Can you see my
yes,
presentation?
yes. Your screen is visible. Yes, you can continue with the presentation.
Please also tell me whether slides are moving. Is it moving?
Yes, yes, it's
Okay,
moving.
all right. Yeah. Thank you so much for giving me this opportunity. And actually, I feel that COVID pandemic has given us beautiful opportunity that we can interact to the scientists in the similar field globally without visiting to each other. And this is one good thing which I can make a note of it. And that is how I could see that Busra is there when I have been thinking that we should meet each other, but we could never do that. So thank you for making such platforms also for online presentation. Uh, can you hear me clearly?
Yes, we can hear you clearly.
Right, right. So the topic which I have selected for presentation is DNA damage response mechanism in Deinococcus radiodurans, and this bacterium. I hope that microbiologists would have heard the name of this bacterium. It actually looks like it exists in tetrad form. It's a red pigmented gram-positive bacterium, and unlike other most of the other bacteria, this bacterium has several unique cytogenetic features, and one such thing is that it contains multipartite genome system where more than one chromosome and couple of plasmids exist in this bacterium as a part of its genome. And another interesting thing which I am sure microbiologists would be interested to hear, that unlike most of the bacteria, this bacterium has ploidy in its genome elements. The number of copies exists 4 to 6 copies or goes up to 10 copies per cell, which is quite unique for this bacterium. This bacterium is actually known not for these cytogenetic features, but it's known ordinary resistance to gamma radiation. When we compare in this slide, there are two panels, left and right. The left side panel is actually the gamma radiation response of mammalian cell, human cell. And when we see y-axis and x-axis, the radiation dose, it goes in the centigrade. Centigrade means 2 gray, 4 gray, 6 gray, 8 gray, and 10 gray. And for human cell, the D10, that is the dose of gamma radiation that can kill 90% of the cells, it turns out to be 5 gray. But when you compare the gamma radiation response of human cell with the bacterium, I mean this a whole lot of bacteria on the right hand side panel, you see that they run in kilograms, and several members of Dinococci family, the D10, that is a dose of gamma radiation that can kill 90% of bacterial cell, it goes to 10 to 15 kilogray. Now you look from 5 gray to 15,000 gray. That's a kind of gamma radiation resistance. The several bacteria belonging to Dinococci family, they have it. Not only that, this bacterium also has got resistance to several abiotic stresses and DNA damaging agents. So we got curious around 22 years ago, after the genome sequence of Deinococcus radiodurans was published in 1999, that what contributes to the radio resistance, extraordinary radio resistance of this bacterium. When you look the next slide, when the DNA damage and repair kinetics was studied for this bacterium, after giving 6.5 kilogray gamma radiation, the genome of its bacterium gets fragmented to around 25 kb. The million bases goes to 25 kb. It produces nearly 200 double strand breaks. But if you leave this bacterium in the same medium, then a little less than 8 hours time, it actually resumes the patterns of the unirradiated cells. This was, I'm sure we all know that bacterium like E. coli or bacilli, they cannot tolerate even 4 double strand breaks. And here is a bacterium that can tolerate 200 double strand breaks without losing the viability at this dose of gamma radiation. When you check the fingerprint of the genome by looking the NOT1 pattern, what we observed that in 8 hours or 20 hours time, you can perfectly see the complete resumption of the NOT1 pattern of the unirradiated cell. So scientific communities around the world, they jump to the conclusion that this bacterium not only having an efficient DSB repair mechanism, but this also retains its original genome without any genomic rearrangement. And we started working on this bacterium by addressing a large number of questions which I am not going to cover here. But I want to draw your attention that gamma radiation, which is actually ionizing radiation, it produces harmful effects by two different mechanisms. One by direct deposition of energy on the covalent bonds, and it breaks the bonds of the DNA and protein oxidation, etc. The other mechanism by which ionizing radiation can kill the cells indirectly through production of reactive oxygen species from the radiolysis of water. And this is the whole lot of mechanism where you see superoxide radical, hydrogen peroxide and hydroxyl radicals. All these deleterious radicals which can be produced by radiolysis of water from gamma radiation exposure, they can actually damage the protein, lipids and nucleic acid. So when we started addressing this question, our first concern was to understand that how come this bacterium can tolerate such a high doses of reactive oxygen species, and what is the mechanism of oxidative stress tolerance. That's the area which I am going to concentrate in next 10-15 minutes time. So the cut story short, after 5-6 years of the work, this slide actually summarizes where we identified a compound from this bacterium that is called pyrroloquinoline quinone, and this compound was before known as a cofactor for gluconate dehydrogenase and was implicated in the mineral phosphate solubilizing phenotype of bacterium. But in 2003, ours was the first report where we demonstrated that this compound, if you make in E. coli, it can make the E. coli more oxidative stress tolerance, particularly against photodynamic effect of rose bengal. And then subsequently we showed that PQQ, I'll name it PQQ subsequently, please bear with me, pyrroloquinoline quinone abbreviated to PQQ. We subsequently showed that if you take the PQQ in the solution and do the pulse radiolysis, produce whole lot of ROS species, it can actually neutralizes all the reactive oxygen species in the test tube. And there we defined that this compound can act as an antioxidant, and at very high concentration it may act as a pro-oxidant as well. And this work was published in 2004, a few years later. Our interest was actually to provide the all kind of exploitation of this particular compound PQQ for whole lot of dreams which we had those days. But what we did, that if you see in this particular slide, there is a gene is called PQQE gene, and in bacterial system, the PQQ can be synthesized by involvement of 5 proteins, that is A, B, C, D, and E. And PQQE is actually the terminal and regulatory enzyme of PQQ biosynthetic pathway. So what we did, we deleted PQQE from Deinococcus radiodurans and checked for the gamma radiation resistance with the anticipation that if PQQ is involved in oxidative stress tolerance, this might give a moderate sensitivity to the organism. But what we found to our surprise, that this mutant which does not make pyrroloquinoline quinone or PQQ became sensitive nearly 3 log cycles at 10 kg of gamma radiation as compared to wild type. When we check it, the DNA repair characteristics of this mutant, what we found that while wild type can do the reassembling of the shattered genome very well, but this mutant which does not make the PQQ has lost the DSB assembling ability. So there we actually said that PQQ works in this bacterium possibly beyond its role as a oxidative stress tolerant, that is as an antioxidant. We started looking to provide the evidence for that. So when we look the Deinococcus radiodurans genome encoding protein in search of proteins that can interact to the PQQ, this idea came from gluconate dehydrogenase, because gluconate dehydrogenase, methanol dehydrogenase, these were the enzymes whose crystal structure is solved, and the site for PQQ interaction was already demonstrated. So by using that information, when we check the proteome of Deinococcus radiodurans genome, we found that there are 5 proteins in Deinococcus radiodurans. They have varying number of PQQ interacting motifs, that is beta propylar structure. And this green symbol is actually indicate the site where PQQ could have interacted to these proteins. But none of these proteins were characterized before that. So when we found that there are 5 proteins to which PQQ can interact, we went deleting one by one from the genome of this bacterium, and we created 5 deletion mutant, 1, 2, 3, 4, and 5 separately. And when we check the gamma radiation response of all 5 mutants, we found that rest of other 4 mutants, they show the gamma radiation response very similar to wild type. But one of the gene, when we deleted DR2518, the cells become hypersensitive to gamma radiation. And this particular mutant has also lost the DNA repair capability. So from there we concluded that DR2518 deletion made this bacterium hypersensitive to gamma radiation, and that possibly works through inhibition or abolishing the DNA repair capability of this bacterium. We did extensive characterization of this protein. To cut this story short, what we found that this protein has the 7 PQQ binding motif. If you model all this particular C-terminal region of this protein, it actually models like this, and PQQ can be docked in the beta propylar structure of this particular protein. And by doing extensive studies for several years, we characterize DR2518 is characterized as a radiation responsive serine threonine tyrosine type quinoprotein kinase, and we named it RQKA. And now this DR2518 started being called or known or identified in the global literature as a RQKA. Now this protein, since it was a serine threonine type quinoprotein kinase, so we thought of checking whether Deinococcus proteome has got proteins that could be the substrate for this kinase. And what we found that there are 46 proteins which actually have a signature, conserved signature, where the serine threonine protein kinases could transfer the phosphate, and the most notable ones happens to be PPRA, I mean DNA repair proteins on this side, then cell division proteins, including even FTSZ and other things on the other side. We checked in vitro study by incubating with the purified kinase with some of these proteins, and we found that REK, PPRA, and FTSZ, the three prominent player of cell cycle regulation in bacterial system, they are getting extensively phosphorylated by this kinase. We move subsequently to characterize the role of, to check whether phosphorylation can it make impact on the activity of these three proteins, which I am going to show you in next couple of slides. Excuse me. So what we did, that we knew before that PPRA, one of the protein which just now I mentioned here, this particular protein was already characterized by Japan Atomic Energy scientists. So we knew how to check the function of this protein. What was shown earlier, that PPRA can stimulate the DNA end joining activity of DNA ligase under in vitro condition, and this paper was published in Molecular Microbiology. So we took this information and we asked the question that when PPRA is phosphorylated versus non-phosphorylated, how does it affect the DNA ligase activity in vitro? And this slide actually, it tells us that if you have when you have phosphorylated form of the PPRA versus unphosphorylated form of the PPRA, and if you start looking the intermolecular ligation product versus intramolecular ligation product, that is self ligation product, what information you get it, that the moment PPRA gets phosphorylated, it starts making a large amount of very high, in other words, you can say that the kinetics of DNA ligation from intramolecular ligation, self-ligation, to intermolecular ligation has increased when PPRA was phosphorylated. And this was quantified to reach you out that phosphorylated form of the PPRA, it makes a lot more intermolecular ligation, which is tail-to-tail requirement for joining of the DSBs for during the double strand break repair. So, and then we check whether this characteristic holds true under in vivo condition or not. So we obtained the mutant of PPRA, phosphonegative mutant of PPRA by removing the threonine 72 with the aspartic acid. And when we think of complementing the PPRA mutant with intrans by expressing the different variants episomally, what we observed is that wild type could complement complete loss of mutant near to wild type, but this phosphomutant could not do that, suggesting that phosphorylation of PPRA plays very important role in the radioresistance of this bacterium. So based on that, we concluded that when PPRA is phosphorylated, its activity gets modulated, and it becomes useful for this bacterium to confer the higher radioresistance. We also thought of providing another evidence that effect of phosphorylation on REK function. I'm sure we all know that REK is actually a recombinase that does strand exchange activity during the homologous recombination, and equally important for DSB repair as well. So this enzyme REK actually does differential activity when energy source is either ATP or DATP for strand exchange reaction. So we did extensive characterization of this function, but for convenience and not to messing up with lot of data, I am going to show you one slide strand exchange activity of DRK when it was first phosphorylated versus unphosphorylated under two conditions. In one case, when ATP was used as an energy source for strand exchange activity, in other case when DATP was used. And as it is quite apparent that when ATP was used as a for strand exchange reaction, then unphosphorylated form of the RK can do the better activity as compared to phosphorylated form. But reverse is true. When ATP was replaced with the DATP, now the phosphorylated form starts showing the better activity than the phosphorylated form. So this actually tells that RK phosphorylation, it enhances DATP preference for its strand exchange activity. That also fits to the model that during post-irradiation recovery, when organism is recovered, that time the lot very massive degradation of the DNA occurs also, and that produce the DATP pool or DNTP pool in the cells increase phenomenally. So there we argued that possibly that when RK is phosphorylated, it becomes efficient for utilizing deoxynucleotide triphosphate for its C activity as compared to normal nucleotide triphosphate. We also check this activity, the effect of RK phosphorylation under in vivo condition to see whether phosphorylation affect the gamma radiation resistance, by the RK. So we had the RK mutant, and you can see that RK mutant of Deinococcus is very sensitive. But again, when we isolated a large number of mutants of RK phosphomutant, when we try to complement by expressing introns, the wild type RK could complement the complete loss of the mutant phenotype, but the phosphomutant could not do to the full capacity, suggesting that phosphorylation of RK is again important for its role in radio resistance, and phosphorylation enhances the preference of RK for the DATP over ATP. So from these two pieces of information which I have shown you, the first time we reached to the conclusion that serine threonine phosphorylation role in DNA repair protein plays an important role in bacterial response to gamma radiation. And till that time, this was the first report to demonstrate that serine threonine phosphorylation has some role in DNA repair in bacteria also. We thought of checking the involvement of RQK in cell division proteins and FTSZ, FTSA, and DU4. These are the three cell division proteins. They are actually getting phosphorylated by this kinase. We characterize the effect of phosphorylation on FTSZ and FTSA little bit, but DU4A now we have completed. And what we find that when we compare the activity of phosphorylated versus unphosphorylated form of FTSZ, as we know that FTSZ is a tubulin homolog of the in bacteria, and it has GTP as activity. So GTP is converted to GDP, and when this activity was monitored, what we observed that phosphorylation of FTSZ actually enhanced the GTPase activity of FTSZ, and the higher GTPase activity actually makes this enzyme less efficient for cell division. We isolated the mutant of phosphomutant of FTSZ, and this is electron microscopic data. I'm sure you will be convinced that if you have wild type and phosphoablative mutant, then the then polymerization does not get affected. But then the moment we convert this these sites serine residues to phosphomimetic form, then this protein loses the polymerization. And the higher activity, as I said, that higher GTPase activity actually it links to the loss of polymerization, and that possibly makes this protein less efficient for the cell division. So what will happen on one hand when we are having, so this is the summary slide of the entire conclusion, that we had gamma radiation exposure that induces oxidative stress and DNA damage, and both of these stresses of gamma radiation can collectively help. In one case production of the PQQ is increased, in other case RQK, a kinase level increases. PQQ and RQK, when both join together, then they convert RQK into active RQK, and this kinase can phosphorylate whole lot of proteins. Some proteins, when they are phosphorylated, like DNA repair protein, they gets activated and they acquire the improved fraction but function. But when the proteins like cell division proteins are phosphorylated, they actually loses their intrinsic function, and that would result arrest of the cell division till the DNA repair is completed, and then again it resumes the cell division. So based on that, actually with a whole lot of work for several years, we reached to conclusion a possibility of serine threonine protein kinase based DNA damage response cell cycle regulation cannot be ruled out in bacteria. This is what we talk about. Why we are saying it? Because in bacterial system, as a microbiologist will know that Lexe and Recce system is only known well established DNA damage response cell cycle regulation in bacteria, because most of these studies carried out by in E. coli and bacilli, they actually concurs this model, that the cell DNA damage, when DNA repair is enhanced, cell division gets arrested, and when DNA is repaired, then cell division again resumes. But what happens that this particular bacterium, which I am referring, Deinococcus radiodurans, this actually lacks the Lexe and Recce type SOS response. So because many of the components of the canonical SOS response of Lexe and Recce, they are not present in this bacterium. So from this, actually we are attempting to conclude and to throw a hypothesis to the world community that bacteria can also have DNA damage response and cell cycle regulation based on the serine threonine protein kinases, which could be very similar to the eukaryotic type, because the eukaryotic DNA damage response and cell cycle regulation is totally regulated by serine threonine protein kinases. This is the slide, a limited list of the colleagues, those who have been actually working with me for last several years on Deinococcus and have contributed in the growth of this one story. Like that, I am having several 5-6 independent story to talk about on Deinococcus. The main contribution what I have talked about is by Dr. Khairnar, Dr. Y.S. Rajpurohit, Dr. Swati Kota, and Dr. Kruti Mehta, and then Dr. Ganesh Maurya. These are the colleagues who have contributed in this project which I am having. And of course, you will find me for last 20 years, I have been someone who is sitting and all the time thinking that when Deinococcus genome gets fragmented, produce a heap of double strand breaks, then I have been thinking that how DSB is being repaired. That has been my primary thing for last 20 years, 25 years. But we do not know exactly that how its multipartite genome system and ploidy can be retained or originated from the heap of DNA damage or double strand breaks, which repair likely to be random process. Then how this bacterium actually attains at the end of it that this will be chromosome 1, this will be chromosome 2, megaplasmid and small plasmid, and that is something which my current interest, and we have a very good progress on that. Thank you so much for your patience. I'll be happy to take a couple of questions in case if there is interest. Thank you so much.
Thank you, Dr. Hari Mishra, for your presentation. So, do we have any questions? Any questions?
Yeah.
Okay, I guess
My
there
one
are no
query.
Thank you.
questions.
Yeah.
Yeah,
Thank you very much,
yes.
Professor Hari S. Mishra sir. Sir,
Yeah.
we are plant pathologist here at IIVR, Indian Institute of Vegetable Research. We are working in trichoderma, and that is the eukaryotic fungi. So this type of
You
is what is serine threonine kinase, like this system also working in the same way.
see where you are
Trichoderma,
in
eukaryotic
IIVR.
IIVR.
Where is this? Where is this located?
This
I'm
is
sorry for my ignorance.
it is located at Varanasi, sir, Indian Council of Agricultural Research Institute, that is Indian
Okay.
Institute of Vegetable Research, Varanasi,
Oh,
Uttar
okay.
Pradesh.
All right. So, right. So, Dr. Gupta, tell me what's the question which you have?
My query, sir, that system, you innovative way and approach using this bacteria, that particular model may be implemented in the trichoderma, that is also means microbe, but eukaryotic fungi used as the biopesticides and biocontrol agents.
Yeah, I think I'm sure you know trichoderma genome sequence is known, and trichoderma has got whole battery of serine threonine protein kinases. We also have one of our colleagues, he is working on trichoderma. We have very good program in BRC on trichoderma. I hope you know it. And there, I think they are trying to understand the developmental regulation in trichoderma, sporulation, etc. But nobody is actually attempting to characterize the this serine threonine protein kinases roles in DNA damage response, etc. Because I mean, trichoderma is not resistance to DNA damage. The moment you give a mild treatment of damaging agents, it gets killed. So this interest comes basically, Dr. Gupta, to those organisms which have got resistance to DNA damage or have got efficient DNA repair mechanism. And if they don't have, particularly in bacteria, it's more exciting because a large number of bacteria now we know they have got multipartite genome system, and many of them they lack Lexe and Recce type canonical SOS response. So if they don't have canonical SOS response, which has been known the only DNA damage response mechanism in bacteria so far, then curiosity goes that how such bacteria which can tolerate very high doses of gamma radiation and DNA damage, how are they coping up with the DNA damage response mechanism? So that point of view, I mean, those bacteria which does not have canonical SOS response, there it would be worth looking if their DNA damage response and repair is regulated by serine threonine protein kinases. Eukaryotes anyway have it. So trichoderma will fall into the category where a lot of serine threonine kinases are known.
Yeah, thank you, sir. Myself, Dr. Tripathi, not Dr. Gupta, Dr. A.
Oh,
Tripathi.
okay. Yeah, but you were presenting, I think, okay, all right.
Yeah,
Using,
emerging
all right.
disease.
Thank you,
Yeah.
Dr. Tripathi. Thank you.
So, any other questions we have?
Okay, thank you. If there are no questions, I'm
Yeah.
glad. Thank you so much for giving this opportunity. And
Thank
it's a
you, Dr.
wonderful
Mishra, for your
platform.
presentation. Yeah. Thanks a lot. So now let's move on to our next presentation. So for the next presentation, it's from Dr. Eva Maria del Mar Vallis from Faculty at the Bioquímica and UNT Argentina, and on the topic respiratory allergy control by probiotic fermented milk intake, a mouse model from weaning to maturity. So this will be a video presentation, I guess. So Dr. Eva, would you like to play the video or shall we play it from our side? Okay, anyways, I have the video, so yes. We'll be playing the video now.
Hello to everyone, and thank you for taking the time to listen to this presentation. Today I will present to you our most recent published work that was carried out during my PhD in Cerela, and it's titled Respiratory Allergy Control by Probiotic Fermented Milk Intake, a mouse model from weaning to maturity. As the title says, we work with a probiotic strain that was added into a conventional food, which is a yogurt. So let's begin with some definitions, just as a reminder. Probiotics are live microorganisms that, when administered in a great amount, they can confer some healthy benefit to the host. And they can be administered alone or in different presentation or as a part of a conventional food. And when we add it to a conventional food, we have a new food that is called a functional food, generally referred to foods that provide a health benefit beyond basic nutrition. So continuing on what probiotics are, we can name a few beneficial outcomes, such as the prevention or improvement of enteric infections, treatment of digestive system disorders, such as Crohn's disease. They can also be used in the treatment of urogenital system disorders. They can prevent cardiovascular disease. And what is most important, and it is the focus of this presentation, they can work in the modulation of immune immunity through the innate immune response and also have a beneficial effect in allergy. These two can be related also. So, in this image, we can resume how the commensal bacteria, especially in the mucosal surface of the gut, collaborates in the maintenance of the gut homeostasis. But how do we reach to this without entering in more details? Well, the immune system can mature and evolve over the time. Moderation will be linked to the colonization process in the mucosal cavities that takes place from birth. So in general, we can say that the presence of these microorganisms that make up the commensal flora, or microbiota, directs the moderation of the immune system and consequently will exert an action on the moderation of the immune system in the rest of the organism. So, among the individual effects that the microbiota can offer to the gut or the mucosal surface in general, we can say that it can induce the proliferation and repair of the mucous epithelium. It can induce the production of antimicrobial peptides and mucins, and it can activate the immune response to produce more IgA in the gut lumen. Another important thing that we have to take into account is that there is an intimate communication between all different mucosal sites of the body, even if they are very far apart from each other. So, to summarize the relationship between the microbiota and the immune system, we can say that the establishment of a normal microbiota will lead the immune system to a proper function, and we can divide this function into two main features. That is the installment of tolerance and immune activation. In the case of the installment of tolerance, this will be guided to harmless substances, and the immune activation will be guided to pathogens. In the cases of pathogen entrance, we will have two main responses. A first one that is the innate immune response, which is a general response, and if the innate immune response can control the pathogen, it will lead to an adaptive immune response. This adaptive immune response will depend, depending on the type of pathogens, we'll have two different types. One can be the humoral response and the other can be the cellular response. And the humoral response, which is the production of antibodies, specific antibodies, can be a response driven by T helper 1 cells or driven by T helper 2 cells. So if we go back to the beginning and we say that a normal microbiota establishment directs a good immune system development, when this normal microbiota does not establish because of lack of exposure to beneficial bacteria or bad feeding habits or antibiotic abuse or extreme hygiene measures, we will have an aberrant microbiota. At this point, if we have an aberrant microbiota, we may have the problem that there is no installment of tolerance to these harmless substances. And in some cases, this tolerance is not established, and we can have, as a result, the development of allergy. And allergy responses are mainly T helper 2 responses. All these concepts are together part of what we know as hygiene hypothesis. So how does allergy develop? It comes as an imbalance between T helper 1 and T helper 2 response, increasing normal levels of interleukin 4 and interleukin 13 that will induce the production of high levels of IgE instead of normal regular antibodies such as IgM or IgG. These type of allergies are known as type 1 allergies that includes asthma, rhinitis, eczema, food allergies, and in the worst case, an anaphylactic reaction. So innocuous antigens will act as allergens in a sensitive host, especially those antigens with a protein nature. So when the antigen enters the organism for the first time, it will be captured by a dendritic cell. This is an antigen-presenting cell. The dendritic cell will introduce it into the cytoplasm. It will be processed. Then they will travel through the local lymph nodes to be presented to TCD4 positive cells. And these TCD4 positive cells will differentiate into T helper 2 cells. Then they will be producing interleukin 4 and interleukin 13 that lead all the B lymphocytes to differentiate into plasma cells, producing IgE. They will secrete this IgE, and this IgE can travel through the tissue and remain for a very long time in the tissue because they can be attached to the cell surface of mast cells, eosinophils, and basophils, because these cells have in the surface a high affinity receptor for IgE. So when the antigen enters again in a second opportunity or a third opportunity into the organism, this IgE will recognize this antigen and will lead to a degranulation process of these cells, releasing all the preformed mediators. And these preformed mediators will cause smooth muscle contraction, vasodilation, and tissue damage. And all these mediators will continue the response. So we have an increased response. Knowing this, now we can define allergy as an abnormal, fully adapted and exaggerated reaction of the immune system to substances that are commonly well tolerated. One important thing that I would like to clarify is that most of the treatments that are used for allergies are only symptomatic. There are some immune therapies which are very specific to a particular antigen, but they are very expensive and they do not assure a long-term effect. So it becomes necessary to study other options that can be accessible for the population. And this is why it is very important to use allergy models with animals, because we can approach different stages of life, such as adult mice, young mice, or mothers during and after pregnancy. And this is very important because the response observed for one treatment or another may vary from one model to another. So the aim of our work was to determine if a probiotic fermented milk intake exerts a protective effect in mice exposed to oversensitization when the intake of the probiotic fermented milk begins at weaning. That means mice when they are 21 days old, they are still baby mice. And the intake continues until maturity. And also if the mechanisms that may be involved are similar or not to those that we have already previously reported in adult mice. So, as I said, there are different animal models to apply and that we can use to make approaches to treatment. And in the beginning, we started working with adult mice that were sensitized to global warming when they were 6 weeks old, and they had the probiotic fermented milk replaced for drinking water. And we have studied first what happened in the lungs. With these results, we were able to propose a possible mechanistic model, but this was incomplete, and then we had to study what happened in the gut of these animals so we can complete this mechanistic model. And we'll do these results. Mice with probiotic fermented milk start drinking the milk 5 days prior oversensitization, and then they continue through time until the end of the experience. So, from the beginning, the probiotic fermented milk enters the body and reaches the intestinal lumen, which is the priming zone of this probiotic fermented milk, and they can induce probiotics can induce the expression of dendritic cells that have this special marker CD103. These dendritic cells are very special because they are regulatory dendritic cells and they have a migration ability. So, when we sensitize the animals with the antigen, these animals were ready to respond, but not from the allergic point of view. In the lungs, we found that dendritic cells can move to the lungs and they can move the balance to a T helper 1 response instead of a T helper 2 response. Why? Because we found that animals with probiotic fermented milk had a decrease in IgE production compared to sensitization control, and they had an increase in the IgG production. In parallel, this was confirmed because there was a high amount of interleukin 12 that stimulates T helper 1 to produce interferon gamma, and interferon gamma is a regulatory cytokine that controls interleukin 4 production. We also find a small production of interleukin 10, and we didn't see the presence of T regulatory cells in the lungs. So knowing this, we wanted to know if this probiotic fermented milk had the same effect or not in baby mice. First, before we launched our sensitization model, we did a small experimental model to find out if this probiotic fermented milk had any effect on baby mice without seeing the allergen, and only to confirm if the probiotic fermented milk can act as a boost of the immune system. For this, we fed the animals for 10 consecutive days, and then at day 5 and day 10, we sacrificed the mice, we extracted or purified the intestinal epithelial cells, and we cultured them in vitro to see if there was any production of 2 important cytokines that are interleukin 6 and MCP1, which is known as CCL2. Why? Because interleukin 6 is the cytokine that moves B lymphocytes to clonal expansion and let them differentiate and produce antibodies. And CCL2 is the macrophage chemioattractant that leads the cell traffic, and these cells are the link between the innate and the immune response. So, when we compare the results from mice treated with probiotic fermented milk with normal mice that didn't have any stimulus, we found that at 5 days and 10 days, with 4 and 18 hours of incubation, there was a production of interleukin-6, and there was also a production of CCL2 in the mice that were treated with probiotic fermented milk. So now we can say that this particular fermented milk can act as a boost for the immune system. Knowing this, then we proceed to our sensitization model. So, to induce allergy, what we used was ovalbumin. All challenged mice, with or without treatment, had 3 subcutaneous injections with ovalbumin. Then they were left to rest 2 days, and then they were exposed to overaerosols for 7 consecutive days. After that, at 7 and 15 days post-sensitization, we took samples of lungs, serum, and small intestinal fluid. And also, at 15 days post-sensitization, a group of mice that was reserved was re-stimulated with the antigen using 2 OVA injections, and in the middle, 3 days of OVA aerosols, and they were sacrificed 2 days later. So, animals that we used were Baldwin mice from 21 days old. The probiotic fermented milk contains the regular yogurt starters and the probiotic strain Lactobacillus paracasei, 2 species, Paracasei CNCMI 1518. And this is how our experimental groups were conformed. We have a normal control that only has a conventional food and water libitum. We have a basal with 5 days of probiotic fermented milk group that has the conventional food, drinks our milk for 5 days, but does not see the allergen. Then we have a no OVA continuous treatment. That means that these animals took or ingested the probiotic fermented milk from the beginning until the end of the experience without being challenged with the allergen. And then we have 3 sensitized groups. A sensitization control that's only seen the allergen without probiotic fermented milk intake, a previous treatment that drinks 5 days our probiotic fermented milk with oversensitization, and then a continuous treatment that drinks 5 days prior the oversensitization and then continues drinking the probiotic fermented milk until the end of the experience. So now I'm going to show you what were our results. In the case of the cytokine production in serum, we measure by ELISA the production of interleukin 10, interleukin 4, interleukin 12, and interferon gamma. However, these last 2 were not detectable in the serum of these mice. But for interleukin 10 and interleukin 4, what we find was that previous treatment group had an increase of interleukin 10 compared to all the other sensitized groups, but they were not different from the normal control. In the case of interleukin 4, we found a very interesting result, and that is that the continuous group that drinks probiotic fermented milk without having OVA challenge had a very significant increase of interleukin 4. All the other groups also had it, but this was higher. But this isn't a bad result, because even one interleukin 4 in higher amounts can lead to, or it doesn't lead, but it is a consequence of the allergy development. In this case, it is a regular response to produce antibodies. Why? Because if we analyze this with the production of anti-OVA IgE, we found that only the sensitization control has higher amounts of anti-OVA IgE at 7, 15, and 2 days post-stimulus, and that not happen. It does not happen to the animals that receive probiotic fermented milk. At 2 days post-stimulus, we are talking about mice that are already fully grown up. They have approximately 60 days old. We can see that the production of IgE is higher in the 3 groups, but it's still being higher for the sensitization control. In contrast, we can see that specific anti-OVA IgG in serum was increased in all sensitized groups at 7, 15, and 2 days post-stimulus. Then we analyzed what happened in lung mucosa, and this is a very interesting result too. At 7 days post-sensitization, we found that the continuous treatment group had a significant increase in the 4 cytokines: interleukin 10, interleukin 4, interleukin 12, and interferon gamma. Then, when we move to 15 days post-sensitization, this response is regulated and they go back to a normal result compared to normal control, of course. But what is more important is that the sensitization control starts increasing their cytokine production. When we reach to 2 days post-stimulus, all 3 sensitized animals had the same level of this cytokine. But when we analyze this response with the antibody product, we found that, first of all, and very important, there was no anti-OVA IgE detected in the bronchoalveolar lavage of these mice in any group. But anti-OVA IgG was found in BAL, and it was increased in the animals that drink the probiotic fermented milk continuously, and they were gradually increasing from 7 days to 2 days post-stimulus. Also, we found that no OVA continuous treatment had a very special increase of secretory IgA in BAL. We have to understand this as a very good response because we are increasing the barrier, the immune barrier, and we also increase the secretion of anti-OVA IgG, which in bronchus it is more important IgG than IgA. Then we wanted to study what happened to T regulatory cells in the lungs, and we found that for normal control and basal group, there was a small expression of these cells, which is approximately 3%, but this expression falls down when they are sensitized. So this is in agreement on what we found for adult mice, because they didn't have the expression of these cells either. Finally, when we study what happens in the gut, we see that there are no major movements of cytokines or the production of antibodies. As we can see for interleukin 10, all results are similar to normal control and basal control for all groups. Then for interferon gamma, we have a slight decrease, but they are all in the same proportion. Then when we see interleukin 4, as we already see it in the serum, interleukin 4 it is increased in the continuous treatment without OVA, but this isn't a bad result. And we have no special movements of secretory IgA. So what we see here is that baby mice that start challenged when they are young and they start drinking the probiotic fermented milk at a young age, they will have a T helper 2 response, but it is not an allergic response. It is just a normal T helper 2 response for a foreign antigen. So, to summarize, early administration of probiotic fermented milk starting at weaning induces a mild activation of intestinal epithelial cells, preparing the immune system to respond to a harmful external stimulus if necessary. Then, at the systemic level, probiotic fermented milk promoted anti-OVA IgG production instead of anti-OVA IgE production. But remember that IgE was higher in the serum of the sensitization control group. High levels of interleukin 4, which is a T helper 2 cytokine, are related to the IgG production and not IgE, which was regulated by the interleukin 10. In the case of lungs, probiotic fermented milk intake improved total secretory IgA production in no OVA continuous treatment. After our challenge, anti-OVA IgG was significantly increased, as well as interleukin 10, 4, 12, and interferon gamma. In continuous treatment group, with a reduction of secretory IgA. These results show that probiotic fermented milk reinforces the humoral response. In agreement to our previous results in other mice, there were no Treg cells expressed in treated and or challenged pups. And finally, in gut, the probiotic fermented milk recovers secretory IgA levels after OVA sensitization, maintaining the production next to normal values at 2 days post-receivers. There was a slight balance toward a T helper 2 response, but to favor IgA production without altering the mucosal immune homeostasis. We can say then that probiotic fermented milk intake maintains the gut homeostasis during sensitization process and regulates the immune response after challenge, inducing anti-OVA IgG instead of anti-OVA IgE. However, this regulatory effect is not due to a shift from a T helper 1 to a T helper, from a T helper 2 to a T helper 1 response, as observed in previous work, but to a T helper 2 response controlled by small amounts of interleukin 10, avoiding B lymphocytes from switching to IgE synthesis. Thank you very much. I'd like to say thanks to Dr. Gabriela Perdigon, Dr. Carolina Maldonado Valdeano, and Ricardo Whale, which were my directors during my PhD. This is my former laboratory in Cerela, my friends from my former laboratory in Cerela, and this is Dr. Perdigon, and this is Carolina Maldonado. Thank you very much.
So, yeah, thank you for the video presentation, Dr. Eva. So,
Thank you.
for any questions, Dr. Eva is available online. So, yeah, you could shoot them. Any questions?
Yeah. The probiotic microorganisms, those are available in the cats or maybe some other animal products, can be used by some allergic persons. Those are glycogen tolerants or maybe suffering from some special type of sugar molecules.
I'm sorry, I have trouble hearing you. Can you please repeat your question?
These probiotics are beneficial for human beings and available in cats like animal products. Those persons are suffering from carbohydrate means sugar allergic. They can use of this type of bioproducts.
Um, it is very hard for me to understand. Can you write your answer, please?
Could you please just type the question in the chat box so that Dr. Eva can just look at it?
Please.
Okay.
So, in the meantime, do we have any other questions? Oh, we are sorry, Doctor. We're not able to hear you, Dr. Kuachi.
No, I'm not able to hear you either. I'm waiting if the first question is, can you type it please so I can read it and then I can answer your question?
No, we are not able to hear you, Dr. Kuachi. Maybe could you please try connecting or disconnecting the headphones to your system?
Well, meanwhile, I'm not sure if I understand well the question, but
What we use is a commercial probiotic fermented milk. Uh, this is able for human consumption. I don't know if that's how the question goes.
Dr. Tripathi, will you be typing the question now or you'll be sending her an email for that? Dr.
Please,
Kuich, we are not able
doc,
to hear you.
Dr. Tripathi, is it right if I pronounce it that way? Can you please type your question so I can answer and I can share it to the other attendants?
Okay, Dr. Eva, you have a
No,
question from.
no. I'm sorry. We haven't done any sequencing study in the gut. We did a small, but we only count general populations, but there wasn't any changes in the general populations in the gut. So we were suggested if we could do it, the sequencing, but we weren't able that time, so we didn't study that. This is a study made in baby mice, but I like to clarify something. In adult mice, we didn't perform any sequenciation, but in the plate counts for general population, we did found that lactobacilli population was increased and the enterobacteria was decreased, but we didn't identify a species. Yes, they can be used in this case, as you say, for lactogen, because this is a probiotic fermented milk. It has already been transformed by by the bacteria, so we're not taking regular milk or fresh milk. So yes, it can be drink. We did some other researches that were not published in using the when we have the complete food. What we did was a centrifuge and we extracted the supernatant so we can see if the part that has not contained the probiotic has a good effect, and it is also good as well as the complete food. And then we study the bacteria alone when we mix it with drinking water, and the bacteria alone they have also a good result. Yes, FOXP3s, we studied in the T regulatory cells. They were CD4, CD25 FOXP3 positive cells, and we have practically no expression of FOXP3.
Yeah, thank you.
Thank you. If you want, I can show you. I have the presentation right here. You can see. I can show you the
Okay,
graphic
yeah,
again if
let
you
me
want to.
just make you the presenter for that.
Okay. Okay. Especially for the FOXP3.
Yes, I have made you the presenter.
Okay, no, I'm not. I can share it yet. Let me see. Oh, there. There it is. Ventana. Let me see. Can you see it?
Yes, your screen is visible.
Okay, now, this is our Treg cells, and you can see here we have the percentage of CD4, CD25 FOXP3 cells. When we gated these cells, we started gating CD4 positive cells, and then we gated among the CD4 positive cells, the CD25 and FOXP3 cells. So we have for basal group and control group, control group and basa group, a small amount that is approximately at 3%, and then after giving the milk continuously or having the oval challenge, they drop the expression completely. But as I said, we do not have FOXP3 cells, but we do have dendritic regulatory T cells. That's what I wanted to show you. Is there anything you want to see or ask me?
I guess so. Yeah, any other questions you have or anything that you'd like to clarify? We'll just check that out. I'm not sure because no one is speaking, so shall I consider
No,
if there are no questions or
I don't know if someone is writing or not.
yeah, even me. Just waiting for that. Dr. Tripathi, do you want to ask any other questions or is it done?
Okay. Okay. Thank you.
Okay. So shall I guess then we can move on to our next session then. Dr.
Okay.
Koichi, are you able
Thank
to
you.
please
Thank you very
talk so
much.
that we, yeah. Thanks a lot, Dr. Eva. No, it's still not audible. Yes, next is your talk. Like, yeah, now it's your talk. So could you please can you please let me know which is the system that you're using right now? The laptop or a PC? Okay, PC. Okay, I guess that might not be an issue. Okay. Have you connected any microphone? Because for some PCs, I guess there'll be two options, like that is one is for the microphone and the other is for the headphones to listen. So I'm not sure if your PC would give the chance to talk without the microphone attached. Maybe could you try that, or maybe could you log in with your mobile by downloading the GoToMeetings app? Okay, yeah. Let me just make you the presenter. Okay, that's okay, no problem. We can schedule your talk tomorrow, not a problem. But if there is any chance you could just check it out, maybe we can give it a try even today, but it's completely all right to schedule your talk tomorrow. Thank you, and we are sorry for the inconvenience. So I guess now we can move on to the poster presentations then. So we'll have the presentation by Dr. Kuichi Ito by tomorrow. So let's move on to poster presentations. And for the poster presentations, we have the next presentation by Dr. Ika Metrivelli from Agriculture University of Georgia, and on the topic regulation of the Herpex lacteus polysaccharide hydrolyzing secretome activity by different carbon sources. So I guess I have the poster I have the poster, so you can just take a look. I'll just display that from my end. Please do go through the poster. Maybe you can just see how it is. For the next 5 minutes, I can display this poster, and you can just have a look at it, and then we'll see. I guess the words letters will be very small, so maybe I guess this is the best way to display so that it'll be easily visible in a bigger size. I guess you all just had, I guess you all had that, like you just had a look at that poster. And for the next presentation, it's from Dr. Margaret Ole. So due to the internet issues, she couldn't join right now. So I guess today that'll be the end. So we are pending with 2 presentations, and we'll reschedule that tomorrow. So let me just inform you once again. So Dr. Koichi Ito's presentation and the poster presentation of Dr. Margaret Ole will be presented tomorrow. And I guess that's all. We have come for the end of day 1, and we'll all meet you again tomorrow at 2 p.m. in British time. So have a great day ahead, or a great evening or night ahead, and stay safe, stay healthy. We'll catch you all up tomorrow. Thank you for this wonderful day with all your research works. Thanks a lot. Have a great day. If you're having any questions or queries, kindly do let me know so that I'll be here to help you out with them. Thank you.